You know what?
Yeah. From here and then input.
Okay. It works? And we are going to see the presentation here because we have the presentation here.
So the presentation.
When Laura shares.
Yeah, when you share.
We have to share here.
Yeah.
Laura ahí.
Sí. Hi, Laura.
Hello. Hello. Hi there.
Hello, Laura.
Good to see you. Lovely talk earlier.
Yeah. Do you see the room?
Yes, I can. I've been watching you all like a silent little spectre.
It's okay.
So, Laura, do you want to share your screen?
Yeah, absolutely.
So when you're ready, you can start.
Hang on a minute. It's not giving me the option. Here we go. Okay, can you all see that?
Yep. Okay. Okay, you're ready.
Okay, sorry, I can't see you guys now when this is sharing, so all I can see is my presentation. So first of all, thank you very much for the invitation to speak today. I have been listening in, so apologies that I've been just like silently watching, but it's been a really interesting set of talks so far. And I'm really disappointed not to be there because I think a lot of the work that my group does has very strong overlaps with the work that's being conducted by you guys in Argentina.
Laura, excuse me, could you put in the presentation mode?
Yeah, for me it is.
It's in present. Okay.
No, probably you have to share the whole screen, not just one of the screens, not just the PowerPoint.
From there.
Yeah.
Could you put the whole screen presentation in that mode? Because here we don't see in the presentation mode.
Okay, hang on a minute.
You have to share all the windows.
Yeah, share the whole windows.
Share full screen.
Do you see this now?
Or full screen.
Now it's working.
No?
You have to do the presentation mode. It's not working. Yeah,
There, yeah?
Is that
Try with this.
presentation mode?
No?
No?
You have to try changing that.
Okay. If not, we present
Let me
it.
try one more time. Hang on.
Because I don't know what happened. You know what happened?
No, share full screen, the whole screen, not just one.
Are you sharing the full screen there in your computer?
Yes.
No, just one window.
No, just one window. The whole screen. It's okay?
You have 2 or 3 options. There is one.
Let me see if this one, hang on, let's see. How does that look?
Yeah, that's fine.
Yeah?
Yep.
Hey.
Good.
Does that change slide?
Yeah, perfect, Laura. Thanks.
Amazing. Okay, fantastic. So yeah, I decided today, because obviously I presented last time, I wanted to kind of just carry on from what I had spoken about in the previous meeting, but then also kind of give you overviews and insights into the research that we're conducting at the moment. So everything I'm presenting is unpublished, and I'd be very happy to collaborate and discuss on any aspect of it. So last time I spoke about FT3, and in particular how it functions as a low temperature short day florogen. And this has been particularly interesting in the UK environment because the full gene deletion means that actually the plants stay sort of more arrested in the early part of spring, and therefore we increase the actual yield potential. Obviously, this isn't of interest for the Argentinian environment. However, there are alleles that actually lead to accelerated plant development, which I thought may be more engaging. And so just to sort of recap on the main findings of that particular project, it's first of all that we saw for FT3 that in winter lines, there was a much higher association to have the full gene deletion. And so it kind of marries up with vernalization, that actually, if the plant needs slower development, knocking out FT3 and having a strong set of winter alleles enables that. And you can see conversely that it's much more present in the spring lines. We then went through and looked at the Watt-seq collection and observed that actually in the Watt-seq, there's a lot of different alleles for FT3 which are not utilized currently. And we could see through looking at those different alleles, different levels of apex development. And so this links to what I was saying, that there are actually natural alleles out there which are enabling faster apex development. And sort of really to confirm this, we went on to generate some transgenic plants. And these are FT3B overexpressors. And what you can see here very clearly is that these plants go through very rapid floral development. So the T0s actually were flowering on the transformation media. But subsequent generations we've been able to grow. And yeah, you can see that under two different temperatures, 10 and 22 degrees, that actually the plants flower much faster, but this acceleration is actually greater under lower temperatures. And so I really think that this is a very promising allele for sort of promoting early spring, sort of when it's still low temperature, shorter day lengths, faster floral development. So that was sort of just to round off what I'd been speaking about previously. And then today, as I said, I really wanted to share with you some of the more recent experiments and developments that we've been working on in the group. None of these are kind of complete stories. And so, yeah, I'm really sharing with you where we are at the moment. And so what we're really interested in working on is floral development, and in particular how plants, and specifically wheat plants, use environmental cues of temperature and photoperiod to time the initial transition from vegetative growth to reproductive, and then move through very clear and distinct apex transition stages, including spikelet formation and floret formation and dieback in wheat. And then finally, the environmental signals which are associated with stem elongation and flowering. And so to sort of start somewhere, I started the group really focusing on vernalization and what we know about vernalization and maybe what we can add to that knowledge. And so in wheat, there's really three very key vernalization genes. They are, as I'm sure you're very aware, VERN1, which is the dominant activator, FT1, which is also called VERN3, which is also very similar to FT1 in Arabidopsis and is believed to be a mobile florogen. And then a gene which is usually just referred to as VERN2. And this is considered to be the repressor of the network. And it's a gene that I will go into in more depth now. So after vernalization, the function of VERN2 is believed to be suppressed, and this enables VERN1 activation, sort of mutually self-activating network to form between FT1 and VERN1 to enable floral development. I should really highlight at this point that it's not just temperature that's important in vernalization. Photoperiod plays a really key role. That's involved both through the long day input into Vern2, but also through the long day flowering pathway through PPD1. And so, as I said, Vern2 is the central repressor in this network, and it's a gene that we've been working on for a while in the lab. And it's actually for a member of a much larger gene family, is often the way. And these genes are called CCT domain genes after the key domain in them, which is the constans-like and TOC domain. And so you can see in this family tree the ZCCTs down here. These are the Vern2 genes, and they fall in with a wider group, including all of the constans in wheat, but also in this tree is PPD itself, and the other pseudo-response regulators, including TOC1. And so you can see that this is actually a family that is comprised of a lot of very important photoperiod genes. So, as I said, Vern2 is ZCCT1 and also ZCCT2. And so it is actually a tandemly duplicated set of genes. And as this is hexaploid wheat we're discussing, there are then 3 copies of these on the A, B, and D genome. The Z in this system stands for zinc finger, which is another one of the very conserved domains that exists within these 2 genes. And so ZCCT 1 and 2 have widely been reported on chromosome 5, and that's actually just the A genome copy. The other ZCCTs are on 4B and 4D. And until quite recently, we thought that actually there was only one of them on 4B, but it turns out that this is just because the Chinese spring sort of genome lacks a fully functional or a fully easily annotated version of the second one. And this is actually also existing and out there in the wheat genome. And so, as I said, the genes contain 2 very key domains. One is this putative zinc finger domain, and the other is the CCT domain. And I think you can see quite clearly that ZCCT1, A, B, and D are fairly similar to each other, and ZCCT2, A, and D are also fairly similar to each other. However, there is quite a lot of distinction between the 2 genes themselves. And so it's this distinction that we started with, thinking, well, are they just acting together in vernalization, or is there actually some neo-functionalization that's actually happened? And so to investigate this, we've done a lot of gene expression, which I won't go through all of it here, but we've also, I think, slightly more compellingly, looked at how these proteins interact in yeast. And so the first thing we could see was quite clearly that they formed different heterodimers. And so every ZCCT gene was not interacting with each other. They were forming different pairs. And you can see here that the 2A version wasn't interacting with anything. So this is the first bit of evidence that we thought, okay, they're not functioning completely in the same way. We then looked to see how these genes interact with Vern1, that central activator in the vernalization pathway. And what we could see was that out of the ZCCTs, the D copies on 1D and 2D did interact with Vern1. However, the other copies didn't really. There was a very small interaction at the highest possible, the lowest concentration, i.e. the weakest interaction. What we thought was particularly interesting was also that this interaction seemed to be very specific to Vern1 allele. And so when we generated plasmid that contained the exon 4 SNP, which is known to be an important SNP for the duration of vernalization in the Vern1 gene, we lost that interaction completely. And so now we're beginning to think that actually, okay, only one sort of genome copy of the ZCCTs are really interacting in vernalization. And actually how they interact is very dependent on that VERN1 allele. And as I said, I won't go into all of the expression data, but I really wanted to share with you something that I think would potentially be interesting for the Argentinian environment, which is the fact that from these different ZCCTs, we have been starting to look at the ones, as I say, identifying ones which we believe are acting in vernalization, but also trying to work out what we think the other genes are doing. And so this is one particular gene out of that family. And we've went through and we've identified 11 different alleles that exist for this particular ZCCT. And what you can see here, that this first allele, which is allele number 1, really doesn't enable survival during frost. So what we've done here is an experiment with lots of different cultivars, and we've treated them to early development frost, which is -10 degrees in our chamber. And then we've obviously ramped it down and ramped it up, and then we've just measured survival. And so score 0 is the plants died, and score 3 is actually absolutely fine following that experience. And so this first allele, the plants either die completely or are very, very unwell. And so we're believing that actually this particular ZCCT version is involved in the frost response. And we're going to start to try and develop nils to actually obviously not nils of the one that caused death, but nils of these other guys, which seem to be very robust in surviving the frost signal. So we think that, as I say, that there's 2 of the ZCCTs which are clearly involved in vernalization. There's another one that we believe is involved in frost, and there's some that we're still trying to work out through use of transgenics. But whilst we've been doing this, we've also been working on other genes that we think are very clearly showing a vernalization pattern and a vernalization response. And from these, we have quite a large family, 15-member family of genes, which are Madsbox domain genes. And the reason I want to highlight these is because that there's one particular copy of these genes, or one particular version of this gene, which is carrying a very large intron deletion, which is obviously reminiscent of what we see in VERN1. And it's when lines carrying that large intron deletion, we see a very clear increase in the expression in winter wheat. And so again, we think that this particular allele is involved in the duration and requirement of winter or vernalization. And therefore, we're also looking into the converse spring versions, which may be another option to slightly fine-tune the spring response rather than having the really dominant effect of VERN1. So, oh yeah, and I just wanted to share with you some of the transgenics for this particular gene, because they're also clearly involved in flora identity, which again is very similar to VERN1 and the full genes. And so we think that, yeah, this does seem to be something that is in that same pathway. Okay, so then just in the second part of the talk, I really wanted to go back to these CCT domain genes and actually focus on PPD itself. So obviously we all know how important PPD1 is in signaling long day signals to the plant, and obviously its role in photoperiod sensitivity and insensitivity. However, very similar to VERN1, it's a very dominant gene, and being able to maybe manipulate the response in a slightly more subtle way would be really helpful for photoperiod adaptation, and also for just trying to control some of the other effects that PPD regulates beyond flowering time. And so to start to try and do this, we really wanted to identify interactors, or at least understand what PPD is doing in the plant. And so to do this, we've generated overexpressors of this gene as well. And you can see here that there was one transgenic line which was really nicely overexpressing, and so we've gone forward with the PPD1 7.1. And as anticipated, overexpressing PPD led to much earlier flowering, and it led to much earlier flowering under long day and short day conditions, which is obviously reassuring for everything that we know. But interestingly, what we also observed was that overexpressing PPD1 actually led to gaining slightly larger grain under long day conditions, but we didn't see any difference under short day. And so this is obviously interesting because the amount of PPD seemed to be relatively similar under those two conditions. And so we've started to ask, how is the PPD gene actually regulating this aspect of plant development? And so to do that we obviously went on and phenotyped everything in detail, and we could really see that actually PPD did seem to have a stronger effect under long days, even though we still saw a height and a gloom effect under short days, and obviously the flowering effect. It didn't seem to impact spikelet number in the same way as it did under long days. So we began to think that, okay, PPD is clearly acting as a strong signal, and that signal is somehow being interpreted differently under a long day environment to a short day, which is sort of counter to what we know of PPD, because obviously when it's more highly expressed in sensitive lines, then it kind of bypasses all other signals. And so to investigate what was happening, we actually went and had a look at how, like in it, we basically decided to have a look in roots for expression. And the reason for that was we were reasoning that in roots, we take away all of the other photoperiod-based signals. And so we could start to see the genes that are almost truly affected by PPD. And so for this, we decided to look at the FTs, and the reason being that we know that PPD1 regulates FT1, and the other FTs, there's a lot of them in wheat, and so there's actually 12 just listed here. And we were wondering, okay, does PPD also affect the expression of these other FTs? And I think you can clearly see that FT1 and 2 behave very similarly, which is as we may expect. But then some of the other FTs, for example, FT3, which was the one I mentioned at the start of the talk, doesn't seem to be influenced at all by the presence of PPD. To further investigate what was going on, we also looked in some other tissues, and interestingly, we started to observe that actually overexpression of PPD sometimes led to the activation of a gene, such as FT2 here in seeds, but also it could sometimes lead to the repression of that exact same gene. So here's FT2, the blue bar being the overexpression line, sorry. And this is overexpression in the gloom tissues. And so in seeds, more PPD leads to higher FT2. In the gloom, it leads to less FT2. We wanted to try and validate this a little bit more. And so to do this, we used the neuroisogenic lines, which exist for the PPD gene. And so I'm sure you're familiar with this, but SI means single insensitive, TI is triple insensitive, WT wild type, and TK is triple knockout. And once again, we went and measured the FT genes, and you can start to see that exact same trend. So in FT2, when there's triple knockout, so no PPD, we see a massive increase in the gloom for its expression. And so really highlighting that actually, yes, in the gloom tissue, PPD1 must be repressing FT2's expression. And then we went on and obviously looked at the other FTs that we were also interested in. And once again, we could see that these are showing a very clear developmental stage, but also tissue-specific activation or repression by PPD. And so going forward, we really want to start to understand how PPD is both the activator and the repressor. And our hypothesis would be that actually PPD is binding to other components to enable this. And so we really want to identify what these other components are and then use that to be able to have a much finer regulation on the activation or repression of PPD in different tissues. So that's pretty much everything that I was going to say. So just to sort of summarize what we're doing going forward, we are continuing to look at the ZCCT's function in wider plant development, and also the function of those MADS-box genes in spike development and floral identity. And as I say, if any of that is of interest to anyone and would like to collaborate, then please do get in touch. So just to finish on highlighting the people who have actually done the work. So Dom Hirsch has done all of the VERN2 research. Katie O'Connor is working on the MADS-box. Indy, I didn't really get to talk about her work today. She's continuing on the speed vernalization work that the group's been doing. Davina Song has been doing the FT and PPD story. And Pablo Gonzalez, I also didn't get to talk about his work today, but he's doing some stuff on the DSW project. So yeah, thank you. And if you have any questions, please ask.
So questions? If someone have a question, please come here to ask Laura. One question.
Okay.
Hi, Laura. Fernanda.
Hi.
How are you?
I'm good, thank you. How are you?
Hi. Laura, I don't see if you, I don't remember if you show it, the overexpression of the PPD1, your transgenic line. I don't remember seeing the phenology. Do you have difference in phenology in the duration of the phases? Because you show
Okay.
and you show the expression in different tissues, but do you have differences in the time when you have the air, terminal spikelets, and those things?
Yes, so it is. It's basically racing through development. So everything is faster. Yeah.
Well, okay, that's well, because maybe the reduction of the differences you are seeing in the chillers and those things are associated with the reduced duration of the phases, or maybe I don't know.
Yeah, no, absolutely. And I think, yeah, so that's why we were particularly surprised that the grain seemed to be bigger, because we would have thought that it was going through everything much faster. But I think there is some split, that maybe increasing PPD, you actually then somehow are also increasing a repressor that's normally turned off in grain filling, or, something like that. So, yeah.
Okay, well, really interested.
Thank you.
Bye-bye.
See you.
Hi, Laura.
Oh, hello.
I thought I'd ask a question just so you could see I was here and I was listening.
Very good.
You mentioned about the FT3 alleles that you'd seen in WhatSeq. Most of the people who don't know what WhatSeq is.
Ah, sorry.
You might just sort of explain that, and then maybe you could just tell me, a quick summary of what you found in terms of different alleles or what significant different alleles is. Thanks.
Sure. Sorry. Yes, I forgot. This WhatSeq collection has been going. I think it's been something that we've been thinking about a long time now. So I forget that it's not out there yet. So the Watkins collection, which has been published previously, was or is a collection of landrace lines from the 1930s, and it's there's about 800 lines involved in this collection, and they are worldwide. So there was a civil servant who went around and collected them all. And in addition to those sort of pre-Green Revolution land races, there's also around, I think, 500 modern varieties which have been added. And these have all been resequenced using short read sequencing. And this particular resource is just in the pipeline for being made public. And so that's a project that's been really led by Simon Griffiths at the John Ennis Centre. And for my group, we've had early access to the sequencing. And so that's where we've seen all of these different FT3 alleles. And so the alleles that we observed were obviously the deletion, which is very prevalent. And then there were two other alleles, which both were effectively like the deletion. So there was a missense, which caused effectively a knockout of the gene, and a promoter deletion. And these were prevalent in the modern germplasm and had been selected for in modern lines. And then there were all of these other alleles which seem to not really have been selected particularly prevalently in modern varieties. And of those, there's one which I think, well, at the moment we're still trying to absolutely confirm it in the lab, but we believe it's actually a mutation in one of the introns. And this leads to the increase in FT3 expression. And so I think that's very important and interesting for warmer environments, and also for places with shorter growing seasons, which really want to push the germplasm through, because having a higher level of FT3 is basically a short day floragen. And so that enables faster growth earlier.
Brilliant, thank you.
No worries. Sorry about not explaining what seek. Yeah.
Another question? I have no questions, Laura. That's okay. But thank you, Laura.
No, my pleasure. Thank you very much. Okay.
So we go on with the next talk. Me afeitaste. Ahí va. Conéctate, no. ¿Quieres el puntero? Okay.
I have the Harry Potter stick here to help me. It's a magic. Okay, I'm Thomas Stricker or Striker, same. I'm going to talk about water intolerance in winter cereals. I will focus on some responses of wheat and barley, and some aspects of shoot and wood physiology, not more in woods, but than shoots. I think that most of you are familiar with global change or climate change, and most of you know that as a consequence of global change, there are more or an increased trend of waterlogging events along the years, more droughts and more waterlogging of higher intensity, at least is expected that. And not only that, here I try to tell you that waterlogging is an important stress for plants also in economical terms. This is the loss of wheat in terms of American dollars. Waterlogging is in red, and you can see that in 4 out of the 7 years, the losses due to waterlogging are pretty similar than the losses produced by drought. So it's important. It's an important stress. That's the idea of this slide. For me, it's very important because I dedicated the last 20 years to this. So I'm trying to convince you that it's important. I hope that I achieve that by the end of the talk. So this is important. Waterlogging doesn't just mean more water, because the excess of water is not the important issue here. We have less oxygen. That's the problem. The problem is not having more water, but less oxygen. Okay? So in a well-drained soil, we have high oxygen, low carbon dioxide, and low ethylene. All three of these compounds are gases. Oxygen enter into the soil, and as a result of respiration, carbon dioxide can go to the atmosphere. The same with ethylene. But when the soil is waterlogged, we have a layer, a thin layer sometimes of water covering the soil, and the diffusion of oxygen into the soils is reduced between 5,000 and 10,000 slower than in air. So the soil rapidly becomes into hypoxic conditions, and depending on the temperature, you can have anoxia in 1 day or 2 days or so. Also, in this aquatic media, carbon dioxide accumulates, and also ethylene, which is an important plant hormone because it triggers a lot of adaptive responses on plants. So the problem is the lack of oxygen for respiration. If you don't have oxygen, the aerobic respiration is compromised, you don't have ATP, you don't have energy, and an energy crisis appears. This is to show you some of variation in shoot, root, and yield responses on wheat, in particular from a review we did a few years ago. And you can see here, this is the reduction. This is not the reduction, this is the percentage of the responses compared to the controls, drainage controls. You can see that the shoot mass has a lot of variation, as well as the root mass, the root, and the grain yield has a lot of variation regarding waterlogging stress. For instance, the median for shoot mass is 67% of the controls, which is a reduction of 33%. For root mass, there is a median of 38%, which is a reduction of 62% from the biomass of roots. And in yield, it's in the middle with 57%, with a lot of variation that is not only genotypic variation, because this is a mixture of a lot. This is a meta-analysis, including a lot of experiments, different waterlogging duration, different genotypes, different environmental conditions, temperatures during the waterlogging, during the recovery after waterlogging. So, but this is at least something that tries to show some quantitative results to you. But we know that the moment of occurrence of waterlogging is very important. Romina, Gary, and Daniel here, they already showed us a few years ago that the critical period for yield, both in wheat and in barley, is 15 or 20 days before anthesis. See? So I'm talking in Spanish a little bit. So, and the main component of that yield that is affected is the numbers of spike per plant in the case of barley, and the number of grains per spike in the case of wheat, which are indicated here in the figure. With that in mind, that it's not the same to apply the waterlogging at early vegetative stages or in late reproductive stages of the crop. We made some experiments, not me, but Rocio and 2 other students, more people involved, but we have them here just to prove my point. Some experiments at plant level that we made those in tubes, long tubes, and also some experiments at micro canopy level in big size of containers, 1 meter size. Yes. So the idea is to show you some results of these experiments. of some of these experiments. As I told you, here we have the early waterlogging, the bar in gray, and the late waterlogging. The early waterlogging was done in a tillering stage. The late waterlogging was done at boot stage, reproductive stage, both wheat and barley. We have here photosynthesis, stomatal conductance, and the intercellular carbon. And we can see that at least in wheat, photosynthesis only decreased following late waterlogging, during the recovery from waterlogging actually. And in barley, this was noticed during the waterlogging period, and the recovery was not so good as occurred in wheat. In stomatal conductance variables, we didn't find any response in wheat to highlight, but in barley we also detected some reduction of the stomatal conductance, stomatal closure, during the late waterlogging, which is shown here. Regarding the intercellular carbon dioxide, in wheat this remained pretty much the same along the experimental period, but in barley we detected a kind of peak by the end of the late waterlogging, which was suggesting us damage to the photosystem 2, PS2. Now we can see here, we are seeing here, the leaf greenness in spot unit, which called the spot measurements of adult leaves of wheat and barley, and young leaves of wheat and barley. As expected, in young leaves, as nitrogen can be immobilized from old leaf to young leaf, we did not detect any responses, but in the case of wheat and in the case of barley, both species, the greenness of the adult leaves were reduced by the end of the late waterlogging. Nothing much happens during the early waterlogging. Or we can see here that something happens, also were reduced, but then the greenness of the leaf was recovered. And here we can see the chlorophyll fluorescence responses, and in the case of barley, this drop in the relations of variable fluorescence against maximum fluorescence is indicating that there was a photo damage that I mentioned in the previous slide. It was the case. These were responses at plant level, yes, from the tube experiments. But when we made some experiments at canopy level, here, micro canopy levels, you have to have in mind, I couldn't attend yesterday, but probably Gustavo Slaffer and others already showed this, that the biomass accumulation is related to yield, because you can multiply the biomass accumulation with the harvest index and get the yield. In this case, the biomass accumulation is the integral of the radiation of the photosynthetic radiation incident on the crops times the radiation interception times the radiation use efficiency. Everybody knows this, are familiar with this. So we measure the leaf area index, which is related to the radiation interception that we can see here. And early waterlogging reduces the leaf area index in wheat, in this case, but it recovered with minimal impact on the radiation interception that is shown here. This reduction in radiation interception reflects this response, but then it recovers pretty good. And in the case of barley, here in these figures, we can see that both early and late waterlogging reduces the leaf area index, compromising the radiation interception in both years, because we made both years of experiments, and with very limited recovery, which is also expected. In relation to the radiation use efficiency, we can see here that early waterlogging slightly affected the radiation use efficiency in the case of wheat, here, both years, but it fully recovered afterwards. While in the case of barley, both early and late waterlogging affected the radiation use efficiency, and with a more limited recovery following the waterlogging made at later stages. So, what happens with the nutrient concentration in shoots? This is another aspect that sometimes is overlooked, and there is not much information, to be honest. But we know some critical levels, according to the literature that you can read here, for nitrogen, for potassium, for phosphorus, magnesium, or calcium, and we can see that for at least for two of these macronutrients, nitrogen and potassium, the levels of nitrogen or potassium in the shoot, which is the concentration of these nutrients in shoots, it's below the threshold that is mentioned in the literature. So it's in flattened plants are below such threshold or critical levels. In the case of phosphorus, sometimes it's below, sometimes it's higher than the critical level. It's not so clear. There is no problem for the situation of for the calcium or for the magnesium. It's not so clear, not many measurements. And also we have what happens here with some micronutrients. In the case of manganese, there is pretty similar the distribution of the points in the case of control or well-drained plants and the waterlogging plants. It's not a problem, to be honest, with the manganese concentration. But in the case of iron, which is clearly distributed to a higher values, in the case of the shoots, sometimes this can add an additional effect to the one that was imposed by the lack of oxygen, I mean the hypoxia or the anoxia. But this occurs only after 10 days, 12 days, 2 weeks or so. During the first week, it's not expected to have in the soil such amount of iron available to be taken up by the plants, by the roots, and finally found in the shoot. But sometimes, not always, because sometimes they are not so well connected, and Jorge knows this, sometimes shoots and roots should work in tandem to cope with waterlogging. Yes, that's an expectation that it's pretty intuitive. In the case of wheat, I hope not to confuse you because I have some of wheat and barley, and that's why I highlighted the species in yellow. Here, these authors used the stomatal conductance as a proxy for rat functioning in relation to water uptake. The less affected the roots, the better the water potential of the plants, and they can maintain the stomatal conductance if they can do that. If they can continue with water uptake. And this was the result. One of the main results of this work was the less the roots were affected in terms of root length. That is the variable that we have here, the independent variable. The higher the stomatal conductance, which is the response variable that we have here. This slide is a kind of trick that I put here in trying to connect the shoot and the root part, to start talking a little bit, a few minutes on root responses. There is no much in root responses unless in root function. We have some characterization of root mass responses. On this case, root mass density. It's not so easy to pick message here. I'm not trying for you to look into the details of this figure because it's a little bit complex. But honestly, we saw that both, here we have width here and valley here for the 2 years of the experiment, 2017 and 2018. In both species, the early waterlogging was the one that reduced the root mass density within the first 30 centimeters of the soil, but it usually recovers. It was not the problem in early waterlogging. And the changes in root mass density beyond the first layer, 30 centimeters of soil, are less pronounced. But this is biomass. This is not root function, and I want to be clear with this. We don't have measurements for root functioning. But what roots can do is to adapt, to make some anatomical adaptation to the lack of oxygen, to hypoxia. And the most conspicuous root adaptation to hypoxia or to low oxygen is the formation of parenchyma. Yes? We can see that when a waterlogging event is imposed, the roots have higher porosity or have higher parenchyma, which are gas spaces that are formed in the root cortex. You can see here, this is the result of the accumulation of ethylene and the programmed cell death occurring here, forming this kind of parenchyma lacunae, are called lagunas de parenchyma in Spanish, that works like a kind of snorkel for plants, allowing or showing a path of low resistance for the plant to transport the oxygen through diffusion, or diffusion, should I pronounce. You can see here that seminal roots, or the original roots of the plants, are usually not so porous, has less parenchyma, constitutive parenchyma, but they can increase a little bit the parenchyma upon waterlogging. But the adventitious roots that are the ones forming here close to the crown of the plant, or the zone of transition between the roots and the shoot, or the root-shoot junction, I think it's called, here appear the adventitious roots, also appear as a consequence of the accumulation of ethylene. And adventitious roots can have higher amounts of parenchyma, allowing the diffusive transport of oxygen to aerate the roots, as can be seen here. Here, I talk about this. This is in wheat. I have some on barley. I have some on barley as well. This is a control with few parenchyma in the roots. Early waterlogging was able to produce parenchyma in significant amounts. I think it was 18 or 20%, which was pretty similar to obtain on the late waterlogging. So I want to leave here, but not by presenting you a summary of what I said before, because this is a short talk. So to wrap up, again, to repeat what's the impact on photosynthesis, I think it's not necessary. But my perception on what to look for the different type of waterlogging that can occur, because you can have short waterlogging durations of less than 1 week. You can have long waterlogging duration or events more than 1 week, probably no more than 2 weeks in the field. Probably no more than that, because otherwise nobody is cropping grasslands. And they can occur at early or vegetative stages of the crop, or in late or reproductive stages of the crop. So I will look for early waterlogging and short waterlogging, the materials having high constitutive root arrangement, just to start with arrangement, to start with capacity to transport oxygen to the roots and to aerate the roots and to continue growing. The ability for anaerobic energy productions. There are some genotypes having more ability or are more efficient in fermentation processes. The prevention of reactive oxygen species, which appears after water recedes, the plant or the tissues have to adapt again to the normoxial conditions, following hypoxia or anoxia. Lots of oxygen entering again to the tissues, so they have to adapt again, but in the meantime, there are lots of reactive oxygen species appearing. And under low waterlogging duration, the plant has to make an investment in carbon and in lots of resources, particularly replacing temporarily the seminal root system, the original root system, by the adventitious root system. But this pays the effort only in long-term duration waterlogging. In short-term, no. And also, during long waterlogging events, it's good to see for materials, to look for materials having some sort of tolerance to toxic soil constituents, particularly the ones preventing the entrance of iron into the apoplast and from the apoplast into the cells, because iron can be toxic in their reduced form. Under late waterlogging occurring in late stages or reproductive stages of the crop, a high potential for reserve mobilization to sustain garden filling is something that the genotype should do. And the ability to retain some of leaf greenness, delayed senescence, will also help to do current or assimilation to help with that grind filling, or to continue. And late waterlogging in reproductive stages and long waterlogging start spraying. It's not a condition that the crop can tolerate. So I wouldn't look for this scenario. So thank you.
Very nice talk.
Oh, thank you.
I want to know if there are any information about the intra-specific variability in the font between genotypes.
Intravariation. There are some results of Nico Ciancio that is going to defend his PhD in 22, 23 days or so, 3 weeks. There is some work by Tim Setter in 1990, Annals of Botany, but it's also a compilation of results of 91 or 93 with genotypes, but there is not a large screen that experience at the same time, the same conditions to compare. It will happen.
And the candidate traits are that you could link it with remobilization.
Yes, I think so. That's my view. Maybe you talk to Tim Comer or to Ola Peterson, to all, maybe they can add some others.
Following with the question of Roman, is remobilization is the capacity to produce parenchyma or adventitious or surface adventitious roots. Which is in your opinion, the strategy when you're trying to find differences between
If I think that
genotypes?
yes, it depends on how long is going to be the waterlogging, because the longer the waterlogging, the most useful are the adventitious root system, because it will replace with water absorption and nutrient uptake. Do you mean where the resources are coming to form the adventitious roots, or is
or the velocity or the rate to produce this adventitious root, or the rate to increase the arrangement?
Yes, how quickly they can form a ranking, or how quick they can develop adventitious roots is another try to do. Yes, I agree.
But there are variability in this type of
No, yeah, no.
Okay.
I think that probably is, but I have no data, not only mine, but in the literature to show.
So there is some information about which are the genes that can be controlling, for example, the RNA information.
There is no one gene. Lots of genes are related to arrhythmia formation. And that's the problem to make genetic progress on that. There are other traits, for instance, I don't know, the quiescence strategy that plants can use, not in waterlogging stress, but in submersion stress, that they can put the plant to sleep. It's the sit and wait strategy of the quiescent. There is one gene called SUB1A that controls the percentage of explained variance in 76% of the explained variable is doing that gene. So there is one gene controlling the process, which is the repression of gibberellins in order to keep, do not elongate. Yes, it's not the time. There are genes, but for arrhythmia, it's not possible. The group of Meitsu Su in Tasmania, he discovered some QTLs for arrhythmia formation in barley, but it was so complicated, so many genes involved, that so far, yes.
I have the last one. Do you want a comment about the interaction between the waterlogging or waterlogging duration and the environmental conditions?
Yes. As long as the waterlogging occurs in an environment with higher temperatures, it's much more harder for the plant even to survive. It's very contrasting, the response of the same genotype when under high evaporative demand than under medium or mild evaporative demand. It's completely different the response. So you have to take into account the environment for sure.
But then the oxygen goes down very quickly when you have if you have a environment.
Yes, but in both cases, yes, because you have a higher rate of respiration and a higher temperature. But instead of having depleted the oxygen in 1 or 2 days, maybe in the first day you have depleted the oxygen. So I don't think that explains much. I think that's the imbalance between transpiration and the incapacity of taking up water that make the plants difficult.
One more, please. Yeah. And there are any management strategies to reduce the impact of waterlogging? Maybe not previous, probably later that the waterlogging occurs.
In addition to start praying, no, you have. No, but yes, fertilization after under early waterlogging in order to supply the plants with more nitrogen to speed up the recovery. But it's not always. It's not something that I should or I would advise, because it's not. It doesn't work always. Sometimes you add nitrogen and nothing happens, and lots of times you add nitrogen and nothing happens. There are cases in which adding nitrogen help the crop to recover, and there are others that no. So there is not one strategy that I would not, you should do this and everything's going to be fine. It's not. Even Romania worked with adding nitrogen to speed up the process of recovery, and sometimes worked, sometimes didn't.
Thanks.
You're welcome.
Thank you.
Thank you, Sam.
Yeah. This is a perfect place to start and finish.
Wait 1 minute.
Okay. Right. Well, I sat through yesterday's talks thinking, wow, I'm going to get some questions at the end of this. Because I see a lot of really great quantitative thinking about various aspects of how the crop grows that I'm going to totally blast through here in terms of how the crop model that I use works. You can try and ask me questions at the end, and I'll do my best to answer those. But my focus is on photosynthesis. A lot of what we do in Lancaster is actually about trying to discover phenotypic variation in photosynthesis, but I'm not really going to talk about that today. I'm going to talk about the work that's a bit more in the pipeline of modeling what we could do with photosynthesis on the basis that normally we use that to justify going and looking for variation that we could use in breeding programs, but also in terms of what we would need to do as interventions in order to achieve increases in photosynthesis. So the first part of this is going to be about trying to use models to predict what would happen if we improve photosynthesis in terms of yield in wheat. And then there'll be a second part, which is some potential ways that we could get there. So what I'm going to discuss today is in the context of European wheat. And I'm sure you're all aware of this, but just to remind you, it's a really different context in terms of what yield is. So we were talking about yields here in Argentina in the kind of 2 to 3 ton per hectare range. And in European winter wheats, this is the data from FAO stat, you're often looking at something around about an 8 ton per hectare yield. But what has happened over the end of the 20th century, from where we went from relatively moderate yields through to what we see now, is we've got saturation of yield. So we're in a place where farming and agronomic practices have changed a lot in Europe over the end of the 20th century. And we've got to a place where possibly our genetics and our methods need to change if we want to get anything extra out of this system. That said, the UK holds the world record for yield at the moment, and this chap driving his combine here can manage to get 16 tons per hectare out of a field, probably with some practices that wouldn't necessarily be cost effective under normal circumstances, but he gets in the Guinness World Record book by doing this. So there's definitely an element of capturing that potential yield versus what is generally seen on farms. The reason there's a little diagram here is that this gives a sense for the kind of layout of what is recommended best practice for getting good yield out of winter wheat in Europe. So there'll be sowing sometime in early October. It goes in through the winter and the plants get tillering. And then we get early development in the spring following through. There's a series of nitrogen applications through here to make sure that this crop gets the best growth. The other reason this is in here is that this is how I've parameterized my model for wheat based on the agronomic recommendations. So there are things here about the density of existing nutrients and things that go into the model that I'll discuss in a minute. And I've used this to try and address two questions. So this study was aimed at trying to understand how yield potential in winter wheat in Europe is going to respond to climate change. So a lot of what I'm going to discuss is looking at different climate scenarios in terms of downscaled GCM outputs. And then we're adding on to that question whether or not enhancing photosynthesis would change anything about that scenario. This was all done as part of a project called Crop Booster P, which was an H2020 assessment exercise, set the agenda for what was going forwards in terms of research in Europe. And there are now currently 2 large programs on photosynthetic improvement, at least, going on in the European research area. So what I used is this APSIM combined with what Alex refers to as DCAP. So this is a system that was written by Alex Wu at the University of Queensland. And he basically took Graham Farquhar and Susan von Kammerer's photosynthesis model, built that into a small simulator, and then bolted it onto the back of the existing APSIM model. So my feeling is that most of what we've discussed in terms of modeling so far in this meeting has been in terms of degree day type models that maybe have radiation interception built into them and leaf area index. This goes a step beyond that and makes the leaves into physiological components of the model that respond to conditions as well. So that's all shown in the diagram here. And what they predicted with this in an Australian context, which is where they built the model, was that they could simulate the idea that if they increased photosynthesis, they could potentially have been seeing increases in yield potential of up to about 12%. Now that's only in their best environments in terms of where there's no water limitation. I'll come back to that point later on in terms of how this interplays with different environments. This is also, their study here was done under historical conditions, so this didn't look at what happens in the future at all either. So this is the bit that they built on. You have a representation of photosynthesis, not in terms of these individual physiological pathways and the biochemistry of them, but the biochemical representation of that comes across in the Farquhar and von Kammerer model. So I spend all of my life thinking about leaves in 2 dimensions as this kind of diagram. So we've got net photosynthesis up here, and we've got the CO2 supply into the leaf along the bottom of the graph. And these graphs consist of 2 functions that describe different aspects of photosynthesis. So the blue line here is the limitation that's imposed for the capacity for rubisco carboxylation to drive photosynthesis. And the red line is the capacity for turnover of the Calvin cycle to regenerate the IUBP that feeds into that reaction. Predict those 2 things out, find out which one's limiting under any given condition. In addition to that, we have to think about how the CO2 is actually getting to the site where those things are happening in the chloroplast, so there's a diffusional limitation considered in the model as well. And this model, that's treated in a complex but relatively simple way, in that there is direct modeling of mesophyll conductance and stomatal conductance in terms of how water would move through those different phases. But actually, most of the time under optimal conditions, the model assumes that there will be an operation at CI over CA of 0.7, so that the CO2, the ratio of CO2 transport into the leaf is proportional to photosynthesis. And that gets important when you think about what your interventions could be later on. Okay, I think I probably explained that well enough to go on with the rest of this. So what I had to do in order to get this to work, APSIM had been trained on Australian wheat yields. So it's actually built to do this photosynthesis modeling both for a C3 model in wheat and also for a C4 model in sorghum, in case anyone's ever interested in playing with sorghum. But it's not built in a European context. So I needed to find information about soils, because APSIM has a really detailed below ground component to it. The soil is split into several layers. There's water movement through those layers, the roots explore that volume, there is nitrogen dynamics going on there, there's carbon dynamics. Some of that is shortcut when we do these kinds of simulations where you go over many years, as I'm sure you appreciate. You reset the model to make sure that it doesn't break halfway through your historical run. But soils information was important, and it had to be at the detail where I could actually get the model to work with it. So there's a nice resource called Soil Grids, which actually covers the whole globe, but I was able to get information at the same kind of scale that I needed for everything else from there. I had to get a European wheat in there. Apsin's native list of wheats that it can use are all Australian varieties, and none of those are appropriate to a European winter wheat. They don't have the right level of vernalization requirement in particular, most of them. I had to have climate data, so I got that from the intersectoral modeling project that was available at the time, and that's downscaled GCM output. So this model takes weather as its input, so we need rainfall, we need temperature in terms of daily maximums and minimums, and we need radiation in there as well to drive the photosynthesis component of what's going on. And the final thing that I'm adding in here is some improvement scenarios for photosynthesis. So in terms of training the model to fit with these UK practices, I plumbed in all of these different practices in terms of when there's nitrogen, these nitrogen applications are happening, in terms of when crop was sown, what its density of sowing was, and then tested that against the UK's yield potential trials that are done by the AHDB. So I looked for sequences where I had individual sites in the UK where there were multiple years of yield trial in the same place. Often we don't quite get that. You kind of go between different sites based on who's contracted out to do things. But I managed to find a small number of sites where I had consistent records through the years. And I took the average of a large number of varieties here. I wasn't trying to map onto any particular individual variety, because obviously what's happened through time is that the varieties that have gone into the yield trials have changed. This actually gives a fairly good transect of differences in conditions that wheat could be grown under in the UK. This is quite a large climatic gradient here. And the model does reasonably well in managing to get to the yield potentials that we see at these sites. And my climate inputs consisted of scenarios for CO2 increase. And what we were interested in looking at was this decade around 2050. And in that period, we've got two scenarios. We've got the 2.6 forcing and the 8.5 forcing. You see the red line here is where we're headed at the moment. And that's compared against a set of historical simulations that cover the end of the late 20th century. So I have not published this work yet, and one of the criticisms I got about it was that this is too far in the past at this stage, and they'd like to see something that's a little bit more relevant to now. But from that, there was data available for 4 different GCMs. So each of those will have different elements of error in it. It'll tell you different things about how well the climate is modeled by comparing these different options. And I tack onto that some different scenarios for photosynthesis. So the diagram I already showed you of the CO2 response curve is this. In Apsin, this is based on some measurements that the Australians made of wheat in the field there, and they tuned the Varkman von Cameron model specifically for wheat. You can see that if you change the Rubisco carboxylation rate, you'll push that blue limitation out of the way. That will shift what the leaf is doing under different circumstances. And we can play out increasing Rubisco carboxylation rates, increasing Rubisco and IUBP regeneration, just doing IUBP regeneration, or changing the diffusion of CO2 to the site of carboxylation in the leaf. And this final scenario here combines all of those things together. So these are all manipulations of how photosynthesis looks, but this isn't me trying to change genes in the model. So the way that this is modeled is we look at what the physiology could be like, And we would then bolt onto that in the second part of my talk what we would actually do about the biochemistry in order to achieve those physiological changes. So what I did was I focused this down to parts of Europe where winter wheat is grown. And we had a network, there's a network of weather data that's available for Europe, which had been used in some other studies. So I took those specific locations within the winter wheat growing region as defined by CIMMYT, and then projected out what happens under all of these different scenarios. And the outcomes of this are that in the future, we see increased yields in Europe. Now, that's not really a surprise in terms of modeling. Models suggest that Europe is going to have higher yields in the future, and it's usually a combination of both the CO2 fertilization and the fact that it's going to get warmer. And we discussed the other day about potential for there to be additional disruption on that in terms of extreme events, but in terms of the general predictions of what climate will look like, we anticipate that European yields are going to go up. And then the second part of this question, which is about the fine detail between these different blocks here, is can photosynthesis raise these yield potentials? And so you can see that some of these sets of results here for these different GCMs, which is what the different dots are here, are standing out from the crowd. So everything here is compared against the baseline in the historical case, and wherever we've got a manipulation where the IUBP regeneration is boosted by 20%, then we were seeing increases in yield as a consequence of that. So those are just highlighted in terms of these diagrams. Yeah, so that's the case that improving photosynthesis in terms of this model suggests that we should have a boost in yield. And summarizing all of that, we expect yield potential will keep increasing in future climates, and we expect that increasing photosynthesis should add to that. And the question is, how might we do that? Photosynthetic improvement. So here I just wanted to go over some ideas that are not necessarily that new. Steve Long and Jingwan Zhu did some similar modeling to this about 15 years ago with a generic C3 model. And what this model consists of is a series of kinetic reaction constants for all of the different reactions in photosynthesis. So we have a list of enzymes that are involved in the photosynthetic process, and we have flux and metabolite between those going on in this model. That's parameterized with an original set of protein concentrations that are converted into activities within the model. And then what you can do with that model is you can apply conditions to it and see what happens if you try to say, okay, run me through several generations of selection and see what happens to photosynthesis. So between this set of data with the original parameterization and the optimized model here, there's essentially a selective process going on, where the model chooses to adapt to the different protein levels to get to the best rate of photosynthesis. And what happens in this case is that as your photosynthetic physiologists would expect, you get more Calvin cycle activity, you decrease photorespiration if your goal is to increase overall net CO2 assimilation. The results that you're seeing here are from a specific tuning of this that we did for potato recently. So the original model is very generic to C3 plants overall. What we've done with potato is we've taken a Farkov on camera model specifically for potato, adapted the e-photosynthesis model to that, and then run from there with the model to see what we can optimize. And yeah, so the final point to make about this is, if you just run the model in this form, you get optimization of all of the enzymes in the whole process. Now, there's no engineering strategy that says that we can achieve that under current conditions, because We just can't manipulate all of these 20-odd different proteins all at once. What we can do is we can pick out the ones that have increases, and we can say, okay, is there a set of proteins here that if we increase them, we might expect to see improvement? And the result from both the original paper in plant physiology and this analysis of potato is that if we increase the FBPAs, the SBPAs, and the Rubisco, we should see increases in photosynthesis. And so this diagram shows what happens with the two scenarios that I just showed you. So the original potato model, the optimized model, if all of the proteins are beautifully aligned to give you maximum carbon assimilation. And then examples where if you just increased these three particular proteins that we think would have the largest impacts, what would happen? And we've done that for two sets of CO2 options, one at current levels and one at future CO2 levels, which gives the result that we really need to look at those future levels if we want to make the right choices, because the slight differences in optimization that were targeted at the 600 ppm level did alter how much photosynthetic gain you got from this at different CO2 levels. So that's the point, that for maximum benefit, we should really be optimizing for future CO2 levels. And to tie this into conductance, going back to the results that the Australian study had, when they weren't looking at these fully irrigated situations where there's no water limitation, the benefit from photosynthetic improvement was larger in one particular set of scenarios that they call no linkage between photosynthesis and conductance. The explanation for that lies in how they model what's going on with conductance. So ordinarily, we think of a supply function between the ambient CO2 concentration and this sort of operating point here. And in their model, that's set at 0.7 under good conditions. If you don't break the linkage between, if you break the linkage between stomatal conductance and photosynthesis, what can happen when you increase photosynthesis is that stomatal conductance will also increase. So the reason that they end up with less benefit under these rain-fed conditions from increasing photosynthesis is that the model assumes that the plants are also going to increase stomatal conductance. So in a rain-fed scenario, that's a bad thing because the plant basically uses up all its water supply. So the alternative option that they've got here of no linkage pushes that back to where it should be if the stomata don't open more in response to having more photosynthesis. And just to say that these are all theoretical things that we could dream of doing as photosynthesis physiologists, but these things are actually happening in terms of transgenic manipulation. So there's beam manipulation of the carbon cycle activities that, at least in greenhouse studies, can manage to increase photosynthesis. And there are a lot of people in the UK working on stomatal density modifications that mean that we can also tweak the conductance pathways. Which just leads me to say, these are the various people that were involved in the work I presented here. Obviously, Martin was involved in setting up this interaction. Steve's behind a lot of this modeling work, because I was working for Steve until quite relatively recently. I've been an independent lecturer for about a year. Elizabeth leads the Rubisco research at Lancaster and is part of Malcolm's Delivering Sustainable Wheat project, working on photosynthetic improvement. We've got a set of people who are involved in the Crop Booster P, which included the group in photosynthesis at Wageningen. And this postdoc, Sabrita, is currently working on that modeling work that I described in terms of the e-photosynthesis optimization algorithms. And that's the end of what I have to say.
Thank you for the presentation. I have a question. You consider different photosynthetic scenarios,
Mhm.
and in those scenarios, the changes you are considering all the leaves of the canopy and throughout the entire crop cycle, or just in some leaves?
Yeah, so it's
some
not
areas?
it's not clever enough that we're trying to do different things at different times. The model assumes that any change that's made to the leaf is applied at the level of what the maximums are here. Now, when I present these, I'm giving them in terms of what happens in a leaf that's fully sunlit and saturated and has its stomata wide open. The model is a little bit more clever than that, because what APSIM ordinarily does is it takes the crop growth and development module, and it uses the water availability in the soil to tune through transpiration demand and carbon input what is happening in terms of canopy photosynthesis. So ordinarily, the normal version of APSIM, you've got a radiation term that's telling you how much carbon gain you've got. You've got a transpiration term telling you how much water you can use to get that carbon. What happens in the version with full photosynthesis built on is that then propagates through to what the leaf does. So the canopy is given a budget for how much water it can use, and the stomatal conductance can respond to that. So it's possible for the stomata to close in this model, and what the model ends up predicting under those circumstances is reasonably realistic, so it's like afternoon stomatal closure. The other part of this is that this includes the shade dynamic, so it includes a model of how some leaves are shaded and some leaves are sunlit. And in that case, it says that RUBP regeneration limitation is the thing that's primarily limiting in shaded leaves. And in more recent versions than the model that I've got up here, you can actually access that and pull out the information about those different components of photosynthesis as they go through. Photosynthesis is also modeled on the basis of nitrogen content. So there's nitrogen dynamics built into how the crop grows and develops that will feed forward into whether or not those leaves have a full photosynthetic capacity or whether the photosynthetic capacity is reduced. So it considers quite a lot of aspects, but possibly not the very fine detail.
Does the model incorporate rapid fluctuations in light, and do you think that could be somehow important in terms of photosynthesis of the whole crop?
It's almost like I planted you in the audience to answer that aspect. So the other version of this talk that I give to people is about rapid fluctuations in light. The model considers shade in terms of the Farquhar and Dupuy equations, where it basically partitions the canopy into a sunlit component and a shaded component. That lets it look at what happens with the solar progression, so it can understand at any given time of day how many of the leaves should be shaded, but it has no way of knowing whether an individual leaf has been in sun or shade previously to the time point where it's at. So it doesn't deal with fluctuating light. And a big part of our research at Lancaster has been interested in what happens when we transition between shade and sun, because there is basically a warm-up process that the leaf has to go through to get between those two conditions. And we think that there is something to be gained in terms of managing to improve the efficiency as we go between those two conditions. And we also work with the Wright project, for instance, where they've been looking at the transition in the other direction on photoprotection, relaxation, and trying to maintain as high a possible quantum yield as we go through fluctuating light. So we think that there's something to be gained from that. It's not captured in this model at the moment, and it's quite hard to do. But I did talk to Alex last year, and there is a version of this model now where he has it partitioned into a couple of different layers. He hasn't built it into APSIM yet, but he has the DCAPS version can now do 2 layers of leaves, and so there's a possibility that we could use that to simulate out what might happen to leaves that have had different histories in the past before you got to a particular point. Yeah.
I have one question in relation with your last slide.
The last slide, okay.
Because you comment that there is a group that is working to improve the water use efficiency,
yeah,
reducing the density of the stomata in the leaves. But in my linear reasoning, if you reduce the stomatal density, probably you reduce the capacity, the growth capacity of the crop. And this is the reason that you improve the water use efficiency or not.
Yeah, so are you referring to the idea that if you give the plant more stomatal capacity, you're going to do more transpiration and just basically your flux levels will go up?
Right.
Yeah. I think that situation is context dependent as to whether that is the thing that you really want or whether you want to protect yield under a particular scenario. I think the idea with these reduced stomatal density experiments, at least in the conditions that they do them, is that they've found a range where you can reduce stomatal density without impacting uptake too much. There's also an argument that if you can combine these two things, you can at least maintain, for example, the assimilation rate that you would have with more stomatal conductance, but you can do that at a lower cost to water. And obviously that has the potential to impact what's going on in the soil in terms of movement of water towards the roots and other processes there. But at the moment, this stuff is in greenhouses, and so we haven't got to the point where we can test those kinds of interactions.
I remember John Pachura's work with Richard worked more than 20 years in reduce the size of the vessels in the roots, trying to maintain the water in the soil to improve the water efficiency, but never works.
Yeah, so the classic trade-off is that if you don't have enough capacity to do conductance, then you won't get any carbon in. Yeah.
Thanks, Tom.
Right.
Just lost 2 more people. Anybody else?
That'd be fine.
What's going on?
Yeah, it looks like the list disappeared, right?
Great. My bad.
Good. Okay, thanks very much. Very pleased that you're all still here. I mean, I'd like to thank Daniel and Sam, but Daniel's disappeared, for giving me this opportunity to be here and listen to some really fascinating and interesting talks. Some quite complicated things over the last 3 days. So my presentation will not be so intellectually demanding, but I hope you still enjoy it. And rather than focus on individual traits, what I want to just spend my slot is talking about some of our tools that we've developed. Okay, some of the phenotyping technologies, predominantly high throughput phenotyping technologies that we've developed over the past 7 or 8 years as a requirement for the genetic wheat improvement program that we have. Okay, and of course, I hope that you'll find some interest in some of them, and I hope that there might be some potential inspiration for some collaborations and stuff. I just thanked you, Daniel, but you missed it. for giving me this opportunity. Right. So before I start talking about the tools, though, it also occurred to me that I should say something about our wheat program in the UK. And I could have almost given a whole talk on this. And that's because we have a program called Delivering Sustainable Wheat. And a couple of people have mentioned it already over the last couple of days, including Laura earlier and Sam just a moment or two ago. And this is our major genetic improvement program in the UK, funded by our research council, the BBSRC. It's a 5-year program. It began earlier this year. It's £25 million. So it's quite a lot of money. It funds about 70 or 80 researchers in the UK, distributed amongst all of these different institutes, including Rothamsted, John Innes, Earlham and Quadram. And for these 4 institutes, it represents really important funding to maintain their activities. But then in addition to that, we've got several other organisations, predominantly universities, including Lancaster, importantly, and the University of Nottingham, for example, who each bring in specific expertises. And so in these particular institutes, we're funding 1 or 2, typically, researchers, and enabling them to fund postdocs, for example. And it follows on from a previous program that we've had. So we go through these 5-year cycles. And it's about trying to coordinate the work that goes on in the UK and to avoid unnecessary duplication and actually to enhance synergies. And it's worked quite well. And most of the people who are in the program have worked together now for more than 10 years. And so people know each other, they trust each other, and lots of collaborative work that goes on. And one piece of work that I had Laura just introduce a little bit was the Watkins and the WATSEEK projects. And that's because the basis of this program and the previous program has been about exploiting germplasm diversity, wheat germplasm diversity, particularly going back to older materials to find traits which have not come through modern breeding programs. And actually, so for example, in this Watkins collection, 800 plus landrace lines from all over the world. I did check, no Argentinian ones in the collection, sorry. But representative across the whole of the world, collected in the 1920s. And this has all been sequenced. And we're doing a lot of phenotyping work on it. For about 100 of the lines, we've got biparental crosses. We've made a lot of GWAS panels, etc., NAM panels, and done a lot of GWAS work. So that's one of the focuses. We're also looking at other panels too, different kinds of more modern germplasm. But just as an example, amongst the Watkins collection, I think I've warned somebody that I spent too long talking about the first slides. But amongst the Watkins collection, genetically, probably only 25% of what's there has got any representation at all in the modern germplasm lines. So that's a huge amount of potential genetic variation, which could have all sorts of useful traits. Now, the actual material itself is quite horrible. It's low yielding. Some of it's very tall, subject to lodging, etc. But nevertheless, there are interesting traits, be it disease resistance, biomass potential, nutrient efficiency, all sorts of different things. So lots of potential there. So the idea is to do the phenotyping of this material, select through the material, pull out interesting traits, align that with the genomic sequences, so we can actually then focus down to candidate genes quite easily for the different traits. But then to deliver all of this, and that's what this little bit over here is, to the stakeholders, who primarily are the breeding companies that operate globally, particularly those which operate in Europe and in the UK. So the ones that you know, Syngenta, KWS, Limagrain, etc. And so we deliver the material, we deliver all the results, all of the materials, so any seed materials, derived seed materials, etc., pre-breeding lines, all of the genetic information too, all the phenotyping information, all free of charge and free of IP. So the whole thing, because it's publicly funded, all goes straight through to the breeding companies free of charge. So they love that, of course. But then also, it's not just a one-way flow. They also are involved initially in a lot of discussion about what traits we should be prioritizing, what kind of things we should be looking at, which isn't always 100% helpful, because they just want increased yield and disease resistance. Not really interested in the sustainability traits. But nevertheless, they get strongly involved at an early stage. And then when they get the material and they do some of the initial testing of that material, all of that data from all of these companies, which are normally in competition with one another and are normally very secretive, it all gets pulled together and we look at all this data and evaluate it. And then they make their own decisions what material they want to take forward. And then it goes off into their own private breeding programs. So that's how we've worked for the last several years. And we've tried to enlarge it in this latest program to have a wider range of stakeholders, so more downstream. So millers, bakers, etc., and even the supermarkets. So, and the environmental, and a stronger emphasis on some of the environmental aspects, including working with farmers. So that's what this program's about, and all of the work that I'm going to talk about, which is the phenotyping tools, is all buried within this program, and it's really been about helping us do some of the trait evaluation. So I'm not going to talk about most of it in any detail, but I'm here if you want to ask me. Here we've just got listed some of the things that we're interested in. Yield, inputs, nutrient density and quality aspects. We've got a little bit of work on adaptation to changing environmental agricultural practices, such as no-till, but for the most part it's all about genetics and germplasm. Integrated pest management, interactions with pathogens, etc. There's obviously some work on climate adaptation as well. That's kind of all of the areas that are involved. So a huge amount of work, a lot of people. It's impossible for me to cover it all, but bits of what I will talk about, you'll see fit in those different areas. Okay, so this is the kind of trials that we're on. So some example trials here. This is actually a Watkins set of materials, although I think it's a relatively small trial. The biggest trial I've got running at the moment's got 2,500 lots in it. So it's the 830 Watkins lines in replicate, in triplicate, plus some other lines. And so that's quite a big trial to be working with. And we run that in multiple years. And one of the key things that I'm interested in there is something that's very difficult to do high throughput phenotyping on, is actually looking at the nutrient density of the grains. But some things you just have to do a bit of a slog at. But other things that we look at are obviously yield performance of these different materials, canopy architecture and function, yield components. We try to look at roots too. I'll say a very brief bit about that. We're interested in resource use efficiency, so we're interested in nutrient uptake, generation of yield, etc. Quality aspects, proteins, some of the minerals, again, dietary fiber, soluble fiber, it's also an important quality aspect. All of these things we measure on a lot of this material. We're interested in some abiotic stress work, although not a huge amount. I mean, there is some work currently on heat and drought in the UK. It's not a major issue at the moment in terms of production in the UK. So it's difficult to have a big focus on an area which isn't perceived to be an issue. The resilience is the pests and diseases. So there's a whole range of different pathogens, particularly fungal pathogens, that we're interested in. I won't talk about those today. It's not part of my personal research team. And then we've got some of the climate stuff and some climate modeling, and we've got a good modeler, Misha Semenoff, who's done quite a lot of modeling. I don't know if Sam must know him a little bit. So the challenge we have is to phenotype these sometimes tens of thousands of lines. I've got a team of several people. It's only a team of several people, and when I tell them to go out and phenotype all of this stuff, sometimes they get a bit funny about it. So several years ago, around actually about 2015, we started to try and think of ways we could increase our throughputs. And there are two main platforms that we use. We wanted something that was field-based, because we were strong believers that really a lot of the traits are only really expressed in the field. We want to see how they're expressed in the field. We wanted a lot of, as much resolution as possible, both in terms of the temporal, so the number of time points we could collect, spatial resolution, so we could focus down to individual plants, individual leaves, etc. And obviously we want high throughput. All of those things are almost impossible to get, but nevertheless that was the target. And the two solutions that we came up with, which operate in parallel and still continue to operate in parallel, one is this robotic platform system, which I'll talk about a little bit, and also various drone-based solutions. And we started them both at the same time. This one is a fixed site and can give us high spatial and high temporal data collection. I mean, we can really focus down onto individual components of a plant. And it's all fully autonomous. So we can collect huge amounts of data with this. The drone system is somewhat different, and that's really based on covering those large fields and trying to collect data on those fields. And certainly initially when we started, the resolution was an issue, but technology is improving all the time. The drones are easier to fly now. We don't crash them like we did when we first started. And we've got fancier technology in terms of the payloads. And you can do a lot with the drones. You can do a lot of growth measurements, as I'll show you some data. And in both cases, we can get 3D information, and we can get a lot of growth stage information too. Okay. And there are other ways to go about this, and a lot of people have various mobile devices. And we've tried some different things, but these two solutions work best for us under the conditions that we have at Rothamsted, under the soil conditions and the kind of data that we want to collect. So, just say a few things about the two different platforms and some of their capabilities. And obviously, we can't move this around. I couldn't bring it over to Argentina, Buenos Aires very easily. But nevertheless, you can come and use it if you wanted to. No problems at all. And you can see that it covers a fixed area, and it's fully programmable. The actual crane part moves around the field in 3 dimensions. And all of the sensors are based in, if I just go back one, are based in this box, this white box. And that's what can move in 3 dimensions. So you can go to any part of the field within there, and it can go up and down as the crop grows, or if you've got taller varieties and shorter varieties. And it's got a whole bunch of different sensors in there. It doesn't really matter what the sensors are, we can change them if we want, but they're mostly image-based sensors. So, I mean, you can get a lot of information just from RGB cameras, but we've also got thermal cameras and fluorescence-based cameras in there too, and some hyperspectral cameras. And that gives you the opportunity both for high throughput and high spatial data collection. But then what it does give you is a huge number of images that you need to sort through to extract data. So in fact, probably the key specialists I've got in my team are computer vision specialists who know how to extract features from the images and how to use artificial intelligence technologies to actually extract data from them. And a lot of that work is ongoing all the time. It's all currently focused, almost all currently focused on wheat. Occasionally we look at other crops, but then you have to start all over again. And the thing I like about this system the most is that actually it's non-invasive. You know, you're not pushing something in the field, you're not even walking in the field, otherwise we can get problems of compaction. And the other thing is that it's fully automated. You know, you can program it, you can press the button, and you can collect your whole season's data without having to go back again. In theory. In practice, it doesn't quite work like that. And the reason it doesn't work out like that is just because of the interactions of the weather. And depending on the weather, we'll collect different kinds of data. So if it's very windy, we won't collect data. If it's pouring with rain, we won't. If it's very sunny or nighttime we might collect slightly different to daytime, but it is fully autonomous and programmable and can operate 24/7. So that's the Scanalyzer platform. Just trying to remember what slide I've got next. So here, these are some examples of some of the kind of images that we can collect, and we can collect on a regular basis. And we can have up to 1,000 different germplasm lines, small plots within the area quite easily. And some of the computer vision technologies that we've got have been focused on picking out specific growth stages and quantifying those growth stages too. And the most easy to imagine is the spike counting, for example. And that was something that we started off with, a problem we tried to solve quite early, and that's improved as time's gone on. So some of what I'm talking about has been published. We've published over the years, but then most of the algorithms and the approaches that we've used, we've continued to refine. So what we can currently do is a lot improved on some of the first efforts that we made. For example, this ear counting. So this is an example of an ear counting. We have an image here, and that's the time it took when we did it to process it and to count how many ears there are in that image, with about a 90 to 95% efficiency. And the A4 sheet of paper is deliberately placed there so that we've got something to scale. So, okay, within the Scanalyzer, where we have a fixed position, or if you know the exact height above the canopy, you don't need something to scale. But very often you don't. And so what we've done is transfer this technology into just any photograph that you can take with your mobile phone, or even in some cases images that you can collect from a drone. And as long as you've got something to scale on the ground, you can then work out your ears per unit area, or anything else that you might want to focus on. Okay. Also within the gantry system, interchangeably call it gantry or Scanalyzer, but you know the system mentioned. We have a 3D imagery system, so a couple of laser scanners that each produce point clouds. Fuse the point clouds together, we can get full 3D image reconstruction. The main reason we wanted that was just to measure growth and height. But then we started to think we could get a lot more information from that about the structure of the canopy. And we can, but it's not ideal. And we're looking at some alternatives, and I'll very briefly mention the alternatives in a moment or two. But it is good for measuring height, which seems like a overkill when you could just have a ruler. But if you've got 1,000 plots that you want to look at and you want to look at them every week, okay, it's a job for a student, but it's much better if we collect it automatically and more accurate. At this time of the afternoon, we find a lot of student accuracy decreases. And when we first did the drone work, we did a lot of validation. I mean, you'd always do the cross-checking. And we found that we got some plots that just didn't match up from the drone height measurement and the ruler on the ground. And we tracked it down, and it was to the hand-measured samples that were taken late in an afternoon after somebody had walked around several hundred plots. So there you go. Okay, still with the laser imagery, as I said, we can use that to measure height. This is these are low resolution images. The actual images we get are higher resolution. We've just taken at intervals of a few weeks, and you can see how the crop's growing. And you can go in there, as I said, and extract more information about the canopy. What I actually hope that we'd be able to do would be to look at more detail at the spikes. But in fact, that proves to be beyond the capabilities of this. Okay, but with such measurements, and we can put in algorithms to look at the variation in the maximum height, because you're always going to get some spikes that stick up a bit higher than others, and of course you kind of want to ignore those. So you can put cutoffs and pick, say, well, you want to look at what the 95% mark is for pixels to get your height. And these are the sorts of curves you can get. So here I've got just 6 varieties and some different nitrogen inputs, and this is growth through the season. And you can see in some places there are some bigger gaps, And that was probably when the weather conditions weren't very good. But nevertheless, and this Maris Widgeon is one of the older varieties, so you can see that's taller too. And you can start to look with this kind of data interactions of, in this case, fertilizers with varieties, but you can also start to dissect out some more complex populations, which is in fact what I've got here. So this is looking at some growth characteristics of a population through a season, so just by measuring height, and then taking various specific points and looking to see what QTL you can find at those different points. And you come up with something like this. This is published, so you could look at this, and we can find that there are a lot of growth stage specific QTL, which, okay, you know, anybody who works with wheat knows that's going to be the case, but nevertheless, we can do this quite easily and quite nicely with this technology. Okay. Okay, so I said that I couldn't quite do what I wanted in terms of the 3D imagery with the laser scanners. So we've started using a surface scanner, handheld surface scanner. So this isn't quite the automated high throughput. And at the moment, we've mostly been doing this in the lab, but it's a fully portable system. And with this, we can get much higher resolution imagery, both of the canopy and of the spikes themselves. So I just wanted to show you a few examples of this data. So this is some work that's currently in progress, and you can see these are all 3D images. And what we can start to do, or what we are doing, is dissecting out spikelets, for example, and reconstructing the individual spikelets. So this is all virtually done. So we can measure the size of the individual spikelets. And we're beginning to focus down within the spikelets, okay? And what, of course, we want to really do is to measure the growth along the spike of the different spikelets. What we'd really like to do is to measure the growth of the individual grains. A little bit trickier to do, but not impossible, we believe. So what you can do is, of course, destructively take the spikes and put them through an X-ray CT scanning machine, and you can actually then see the grains in situ. And you can get information on the sizes of the grains. This was with mature grains. We're just working on doing exactly the same measurements at different growth points. And what we'd like to be able to do is to take the surface imagery and the validated X-ray CT imagery and be able to infer what's inside from the surface imagery, which I'm told ought to be possible using some computer vision technology, artificial intelligence. So that's something that's ongoing at the moment, and it's actually looking quite nice. Whew. The other thing that we use the X-ray CT scanning for, which we've used for a longer period of time, and actually in conjunction with the University of Nottingham primarily, is looking at roots and roots from cores. So of all the materials that we grow in the field, well, maybe not all thousands of them, but several hundred of them, we take cores, 1 meter cores. And these cores are then put into the X-ray CT scanner so we can start to look at the root structure in situ. So to measure both densities of roots, but also the structure of the roots. And that's mainly been focused on looking at interactions with soil structure. And this also fits in with what I said about the tilling approaches, the minimum till, and how that affects soil structure. One of the bits of data that we've published recently showed how it's actually the pores in the soil, well, perhaps pretty obviously, but the pores in the soil are the key points which roots interact with to explore the soil, rather than going through the solid lumps. Okay, sounds pretty obvious, but it was still neat to see it, and it was neat to see it in the X-ray CT imaging. And we can now do this also at Rothamsted, just in the last few months. Okay, so quickly to switch to the drones as platforms, and we've had a whole series of drones since we started, as I said, about 7 or 8 years ago. And we've had a whole series because we fairly regularly crashed them, and they weren't that great to begin with. And for example, there are a lot more regulations for GM plant release in the UK than here in Argentina. And so all of the GM trials that we have to do have to be under very restricted conditions. And we basically have a compound. We started off with a really big compound with really big wire mesh You can see where I'm going here. Big wire mesh walls. And we were flying the drones over that, and of course they lost radio control because they were in the mesh. And the next thing we knew, they were disappearing over the horizon. So you learn things like that. And then the other big thing that's changed with time, as you can see, this has just got some regular cameras on board. Slightly modified regular cameras, and they helped us produce a lot of useful information, stereo imagery to look at plant height, but also by modifying the filter systems on the cameras, we could also easily extract some vegetation index information. Of course, things change a little bit, and now we use a much more dedicated device which has got multispectral capabilities, so we can get lots of different indices. And best of all, we've got this very large drone which has hyperspectral capabilities with several hundred different wavebands. And that ought to enable us to screen for all sorts of different features, hopefully useful traits. Not without its problems, but nevertheless what we can. And this also has a LiDAR on board, which I naively thought we could use for measuring crop height, but actually it's much easier using the stereo system. So we still use that, even though it's an older system. And we also have a thermal camera on the drones. I'll show some thermal data, which I put in especially in relation to the talk we had yesterday. I'm rushing so we get to coffee within 5 minutes. Okay, so this is the stereo imagery that we do. So flying two cameras on a drone and then reconstructing the 3D imagery. And from this, we can get very accurate plant height to within a few centimeters of what you'd measure with a ruler. And actually, as I said already, more accurately than with the ruler. And these are the kind of data we can get. This is looking at 25 different varieties, or modern varieties, and again, a nitrogen interaction with multiple time points. And this calibration shows you the degree of accuracy that we can achieve. So this is quite old technology. A lot of people can do this. We can do it fairly routinely. And if someone wanted to learn how to do it, we could teach you the way to do it. Okay, in addition to the height data, as I said, we can also get NDVI data. So this is, again, using a fairly simple, low-cost system to extract for exactly the same experiment, NDVI data. Okay, so once you can start to collect real temporary dense data, you can start to do lots of different curve fitting and start to get parameters relating to the different germplasm. So the examples I've shown here have just been with these 25 modern germplasm lines in a nitrogen experiment, but we can also do this quite easily with large mapping populations or large GWAS panels, etc. And we do that pretty much routinely for all of our experiments. And we've got quite a nice tool. Big issue here is you get all of these images, you have to process the images, but then you get masses of data. So we're putting together simple tools where it can all feed through, and you can get these plots, and you can get whatever parameters you want in an automated way. It's not all quite perfect yet, but that's what we're working on. Trying to be honest. So this is the more recent drone technology I already mentioned with a hyperspectral camera. The advantage of this is you can actually get hundreds of different canopy indices simultaneously. I mean, there's just a few examples here. So it's quite nice, but it takes a lot more processing, this kind of data. And it's difficult to process just the image, the spectral imagery that comes off the camera before you can see anything and before you can start extracting wavelength information. And this goes from 400 nanometers to 2,500 nanometers. So there's a huge lot of possibilities of different characteristics that you can screen for. Okay, so the thermal stuff, we started doing that some years ago. This was our first experiment. This was a mapping population under irrigated and non-irrigated conditions. And you can see what the canopy temperatures look like. And we were massively excited about this and thought this was the bee's knees and fantastic. We still are a little bit excited. And you can see the difference about under the Irrigated conditions, you've got a much wider variation in canopy temperatures compared to the non-irrigated where the canopy is all really hot. There are some complications about this, as it turns out, which in fact was alluded to yesterday, in that you, as well as measuring the canopy, you have interference of the soil too. Okay. However, forgetting that just for a moment, we also found that, depending on the time of the day, you've got very different bits of data. Okay, that's not that surprising for a whole bunch of reasons, both plant-related and air temperature-related, in some cases due to cloud cover. But it just goes to show that, just over an hour or two period, this was between 8:47 in the morning to 3:30 in the afternoon, the latest one, huge variation in temperature. So you need to bear that in mind. But as I said, there is a slight issue here over where you haven't got complete canopy cover, you can also pick up the soil temperatures. So to get over that, we went back to the frame system where we've got this thermal camera, and we can collect thermal images. Well, the thermal images are still going to have the problem of the background and soil, so we did in fact pretty much exactly what you said you did. And in fact, we took RGB images and created masks, and then you could just look at what's going on in the vegetation. That's one solution for doing it. We actually got another solution which we use more routinely now, and that's by looking at all of the different pixels that you get in the thermal camera and separating out the pixels which are going to be soil from the ones that are likely to be the vegetation. And that's actually a better way of doing it, because you don't have to do this matching of the two images. But again, here you can see differences at different times of the day. Again, and then if I extract all that kind of data, this is the kind of data that you do get. So this is over several days and shows diurnal variations due to all sorts of different reasons in the canopy temperature, and does show the critical importance of timing of when you collect the data. And actually you can also relate day-to-day variation to other environmental factors. And we've started looking at mapping populations, including the mapping population that we looked at with the drone using this technology. Okay, I'm there, nearly coffee time. So I do have a summary, even though it's a short talk. First of all, these are high throughput phenotyping technologies mostly. The surface scanning is not quite so high throughput, but you can still get through a lot of spikes quite quickly. But it's all under continuous development. I'm not telling you something here and this is the perfect solutions for everything. And it's all been developed within our week program. So at the same time, we're using the data, it's going in that whole pipeline between the researchers, the pre-breeding material, and the breeding companies. It's mostly image-based, so it does require expertise in knowing what you're doing with computer images. And I've got a couple of really good people who know what they're doing. At least I think they know what they're doing. I personally, I'm not one to judge since I don't know really what they're doing, but I tell them, I want to look at this, I want to find this, and they'll figure out a solution. Usually takes a few iterations. So the computer vision specialists that I've employed, some of them have never seen a wheat plant in their life. Some of them don't know anything about plants at all, but it's a two-way process, and they learn a bit of biology and we learn a bit of computer vision stuff. All these technologies are great for screening germplasm and pallets, which is why we've developed them and put them in place. I'd like to be able to do everything in the field, and we can do everything that I've shown you in the field with the exception of the surface scanning, which we can do in the field and will do in the field next year for the first time. I'm not sure how well it will work in the field, but we've got a high expectation there. In spite of all of the processing of that, you still have massive amounts of data. And our computing at the section at Rothamsted are always hassling us that we've got absolutely huge amounts of data. We're loath to throw away primary data images in case we get better technologies to extract information. And the final thing I should mention is that delivering sustainable wheat as a completely open data policy. I said that everything goes free of IP through to the breeders, but Actually, we've got a commitment to putting all the data into the public domain as an early stage as possible. And we have some websites where that will be perfectly possible. You'll be able to go to a website, pick out one of our field sites, pick out some plots and extract data. That's all on the way and is almost there now. I mean, we can do it internally, it's just not available externally. There is a couple of web addresses here. You can look at Rothamsted and you can find bits of extra information about my stuff. This is my team, sort of half divided it into the people working on the drone stuff. So Andrew looks after it all overall. March does most of the flying, Carol does a lot of data extraction. My name's on all the paperwork if they do crash and kill somebody. It's me who goes to prison, but these guys do all the work. And then the field scan analyzer, Nicholas kind of looks after the physiology. He's a really great crop physiologist. Puria now left and was replaced by Latifa. These are two computer vision specialists. Everything's funded by BBSLC, and it's all, as I said, part of this Delivering Sustainable Wheat program. Thanks.
Do you have any technology or any sensor to take measurements about foliar diseases, for example, or something like that?
If we've got imagery and if you know what a foliar disease looks like, then we could figure out ways to do it. Using spectral information, you can do it too. And one sensor that I didn't mention, there's a reason I didn't mention it, is that we've also got a camera which is focused on measuring chlorophyll fluorescence. And that is also very good for picking up foliar diseases. The reason I didn't mention it is because at the moment it's not working, so I'll put it at the back of my mind. And because of Brexit, I can't get it fixed at the moment, but that's a whole another story. So yeah, you can anything you can see, or if you know it's got some sort of spectral signature, then sure, you can figure out a way to do it. I don't have too many diseases in my trials because we try to avoid them, and we spray every chemical that we can to avoid diseases. But it's possible. You have to pay me a lot if you want to put diseases in my trials. Fernando.
Have you tried to differentiate the different cultivars with the multi-spectral data? So if it is cadenza or fasol or...
No, we've never tried to do that. Actually, that's quite an interesting thought. I mean, we have the hyperspectral stuff on the scanalizer. At the moment.
How many bands do you have? 100? 1,000?
For which? For the
multi-spectral data?
It's several hundred in total for the hyperspectral. Yeah, for both the drone and the crane system. So there are plenty there, and some of them going into the near infrared. Yeah, you could probably try to do that. Never did try to do that.
I didn't show the data because we don't have time, but in the group of HB4, we have people working with image. They work with 1,000 bands, and they could differentiate the HB4 from the normal wheat.
There are some varieties of wheat that are clearly different, you know.
Yeah. But you see, this is different that you cannot see with your eyes. I have my you stand there and decide this is like same color, everything is the same.
It can be the same color.
They can, I don't know, they can.
I mean, structural aspects of the canopy and of the leaves. And if leaves are curling because of some drought thing, you can pick this up, and you can pick it up not just from the RGB images, but actually from the hyperspectral data. And that's because of the way wavelengths are reflected. If you look at them, you probably wouldn't see the difference, but you can pick it up like that. And the other key difference between a lot of varieties, I mean, so the 3D structure, shape of the leaves, and that has a big impact. But then also the waxiness and the wax layers also reflect light, or refract light probably quite differently. And that's why some varieties look much more bluish than other light, for example. So some is really obviously different. Whether or not you could really pick out every variety individually, I don't know.
And have you tried with the LiDAR or the blaster? Have you tried to differentiate purging efficiency, not only spike structure, but purging efficiency itself, just considering the weight of the spike also?
Yeah, I mean, that's what we want to be able to do. And with the laser technology, I don't think it's good enough. And the problem is that I said there were 2 lasers, and we get 2. It's getting a bit technical now. Though we get 2 point clouds that you need to fuse together. And if there's a bit of wind and they're moving around, then it gets difficult. Okay, you can wait until there's not much wind, and mostly we do this laser imagery during the night when conditions tend to be a bit stiller. But it still depends on movement of the crane, and there's just a little bit of vibration in that, which means that if you want to look at fine detail, the 2 point clouds don't mesh together quite accurately enough. So that's why we're looking at a different solution. And we've got these surface scanners. We've got a couple, one of which is supposed to be fully portable.
You showed a figure about the NDVI and I don't know, thermal time or time. So I wonder, how do you transform that information in interesting traits or some traits of your interest?
It depends what you want to measure. So usually we were measuring the rapidity of the canopy formation, the length of time that it's a maximum in dBi, and then the decay as the crop senesces. And so then you can integrate under that and kind of relate how development of the canopy may relate to yield characteristics. It's obviously not as simple as that, as you fully know, but nevertheless, that's the sort of thing you can do. You might only be interested in the early stages of canopy closure, or you might be interested, as I said, in the kinetics of senescence. So different projects have slightly different requirements.
Okay, because there is a good correlation between the NDVI and the radiation interception efficiencies.
Mm-hmm.
You can, by assuming some things, you can estimate bias.
Sure.
With that data.
Yeah.
Your KC, for example. Because if you have this information and match with radiation interception and KC of the crop, it's interesting to calculate the water consumed.
Indeed. Okay. Coffee.
Thank you.
It was great.
Thank you.
Hagamos la presentación.
¿Quién viene? ¿Vos?
Gaby, Román y
Diego.
Ay, Diego, ¿verdad?
Acá está Diego.
Pero de maleza.
Pero
La clase de malezas.
No se maleza.
estamos media hora atrasados.
¿Quién?
Uno, por favor.
Charla Workshop. Asumo bien, ¿no?
Digo yo.
Gracias. Muchas gracias.
Gracias por venir.
Batra es el único.
Gracias por venir.
Estamos media hora.
Lo cortamos acá nomás.
No, hacemos 15 minutos.
A mí es muy cortita. Yo en 15 minutos
No,
tengo
no.
la liquidez.
¿El recuperatorio estás vos sola?
Yo igual puedo sacar algo en el
¿DF?
Sí. Yo en 15 minutos la liquido.
Yo puedo sacar algo en el
Escúchame,
Yo tengo 55 slides.
cortemos el coffee break.
40 slides en la...
Yo tengo que hacer rápido. Encima
No,
mira,
sí.
¿Cuál es Miralles?
No está acá cargada la mía. Yo no está mal.
No, cambió la compu con Jorge y.
Y no dice Miralles.
No, fecha los tipos o nombre acá. Nombre, a ver si. Miralles primero.
Acá, esta, ¿no?
¿Esta?
Esta. Y fija. Un poco largo. ¿No?
Sí, y dormimos en el almuerzo. Ahí veníamos bien y...
¿Qué? ¿Nos atrasamos en
Sí,
el
sí. Y 5 vinimos por acá y hasta que pusimos todo a andar. Nos comimos 10, 15 minutos. Yo sí me estaba...
Yo estaba con mi cabeza. Che, mañana no hay viaje.
Me llamó Nacho, me escribió, dice que había... ¿Lo vi? Acá. Un peor anuncio. Peor que...
Sí, por eso, lo suspendemos, ¿no?
Le dije, mira, lo más probable es que sí, pero déjame que hablo con él.
¿Vos te regresás para Rosario?
Sí.
Mañana.
Sí, aprovecho.
No, te preguntaba por si tenemos que laburar en algo.
No, está bien.
¿Querés tirarla en el
Acá la tiré.
escritorio?
Esta. Esta.
Ah, listo. Perfecto.
Le pongo mi nombre si querés.
No, si no, está la vieja.
Dale. Quería liberar el pendrive.
No, vamos a ver. No, tratamos de recuperar ahora algo. 15 minutos de
Yeah, that's a good response. What we normally have is 3 rows.
No, bueno, buenísimo, ¿eh? Re bien, muy bien. Ya cuando estás andando en bicicleta ya te voy a sacar todo el encima para no llegar al año que viene. Claro, con la mochila muy pesada.
Two places with this big one, 6 more places with small ones with only HCV cameras. And the things, you can use it, Mr. Tom's side, you can use it. Yes? Yes. We're trying to do that.
So, slowly to apply, close to the and any resolution.
Yes, that's it. Yeah. We have to measure that.
So at the moment we can see
Have you tried to do that with here with arms? Because it's like an armless, it works.
all dependent on growth stage. What's better?
¿Sabes si agarro la charla que era? Quiero ver si agarro la charla que era cualquier fruto, ¿no?
Buscarla ahí en el escritorio.
¿Cuál? No veo un carajo. ¿Vos ves eso algo?
Estás liquidado.
Y no tengo anteojos, boludo. Los olvidé.
Arg-Uk Workshop.
No,
¿Cómo se llama? Ah, no la cargaste todavía.
sí, la cargué, pero no sé cuál es.
Bueno, pará.
No, fíjate, ¿cómo está?
¿Cómo se llama? Segunda Argentina. Mejor. Está la mía. Miralles. Uva. 23.
Déjame ver, que después me cualquier cosa y empiezo con una que no es. ¿Cómo se abre esto? Sí, charla UK Workshop.
not everything is difficult to go back to sleep with the noise.
Son el último de Malisa.
I'm going to go 3 more, 4 more.
Chef?
Not 3 more.
Yeah, 2 short. 3 more.
I will.
My thought is
5.
Nice is so just
But
55 slides.
Everybody says, oh, mine's a short talk. Everybody is funny. Mine was a short talk, but sometimes I just start talking about something, you know.
Are you getting the same slide for 20 minutes?
in Spanish probably we've got 1 hour.
Yeah.
In English, that's 5 minutes.
English back then.
Yeah.
Some short.
We can talk a long time too, as well, if we want.
Yeah.
It's perfectly possible.
I sent you the guide
¿Esto
because
anda?
I am leaving
¿Qué?
at
Pasa,
5 more or less. What's the question?
Yo lo sabes lo que tú ibas a la charla. Veo la charla y dices, che, no anda PowerPoint.
No, mi amigo.
No, PDF. Tenía todas las animaciones.
No ando todo mezclado una arriba de la otra.
Y no andaba nada, ni para entender nada.
I can Google it, but it's nice.
Ponele
la cámara.
2 pilas.
Yo no lo tenía.
Really nice to meet you. I will say. Okay. Okay. Thank you. And have 3 hours driving to 5 and come back.
Llego, chino. Muchas veces.
Amigo, no
Una camisa chino, blanca. Digo, eso.
¿cómo se dice?
Digo, si es un político, no le traje un chaco de la película. Sí, a uno le encontraron ahí caminando. Todo lo que pasa.
En esas cosas te lo tienen que avisar.
No es forma, chico. Es todo que lo tienen que avisar. Han avisado, o sea, fui a un lugar donde tenía que dar un deporte, no avisaron nada. Sí, tengo que dar todo de traje, mochila, persona.
Eso es lo peor de todo. Cuando vas sin saber andar.
Ay, tío, no te quedo vestir.
Bueno, yo también me iré yendo.
No hay problema, vos tenés un viaje.
Sí, no, llego.
Pero usted encima se va a quedar a jugar. No, vamos a llegar, vamos a llegar.
No sé.
Hay unos obligados.
Oye, pasa. Yo no sé qué pasa en alto porque está. No sé por qué. ¿Sabes lo que es? ¿Qué me pasó? Escucha, fui a China. Dije, el auto, trabajo. Llego, estaban todos los paraísos terminados en el piso. Terminados en el piso.
Pero mucha agua, ¿no?
No, yo lo cuido. Deberíamos chocar. Claro, no se pregunta.
Yo lo tengo ya, a ver.
Creo
No.
que nunca escuché tantas veces seguidas.
¿Estás aquí?
Ah, esta. No, pero guarda que esta es trigo y cebada, ¿eh? Vos escuchaste
Ah,
trigo por un lado,
trigo y cebada.
escuchaste cebada por otro, ahora la escuchás juntos.
Es un mix.
Ahora la escuchás
Bien,
juntos.
bien, bien. Eso es que está bien.
Igual va a ser doblemente aburrido.
Estamos ya arrancando, ¿no?
No.
Vos qué es lo primero.
¿Por qué no lo limpian a Guille García de él? Ya.
Porque nada, porque trabajó.
Es más bonito.
Se queda un poco de ideas.
Cerrado, vos tenés más cara de colo, vos viste que se ponen las fotos en la
Sí.
montaña. Sí, sí.
Más callo, no te más caro. No te más paquete. Más paquete.
¿Vos ya estás gringo para arrancar? Sí.
Está como medio elevado.
Sí.
Está más
Era-
Más que eso, ese es el palo.
Ah, es en invierno. Es en invierno en la montaña. Ve que ahí está María.
Yo no lo quizás.
Típica foto de colo, vos viste.
No, está bien.
¿Quién es el primero? Vos.
Mirá la fotito que se puso, eso es para dar envidia.
Fue con María, me había dejado a María que. Ok. ¿Qué tenés que comer?
Bueno, la pucha.
¿Qué tenemos?
¿Este? Hola. No, porque se ve ahí también. Se te ve.
Okay. Thanks, Daniel. Thanks, Santiago, for the invitation. It's I hope that it's a short talk. I hope. Well, the title of the presentation is Determinants of Grain Number Responding to Environmental and Genetic Factors in Wheat and Barley. I am Román Cerrado. The working team is Guillermo Garcia and Daniel Miralles from the University of Buenos Aires, and Roxana Sabin and Gustavo Laffer from the University of Lleida in Spain. This presentation is based on two reviews. One of them in wheat that we published on 2022, and another review is in barley that we published on 2023, and I will try to mix both and show you the results. Okay, we were talking about a lot about that wheat and barley are relevant crops, and considering that it's not possible to expand the area for crops, it is critical to continue increasing the yield per unit area. Then, if we have a better understanding of yield determination, we will design more efficient management of breeding strategies to increase the yield. In this sense, we talk about that too. Grain number is a key determinant of yield and is more associated to than grain weight. Then, improvements in yield depends on our capacity to increase grain number. Talked again about that. The most popular approach to understand the grain number and the grain yield is through the numerical components approach. We have two numerical components, spike number and grain number per spike. But there are, there are another physiological approach to explain the variations in grain number and grain yield. The physiological approach is through the spike dry weight and period inefficiency. And this is an alternative approach to use the numerical components approach. grain number response or grain number increments, and the association with the numerical or physiological component depends on the nature of the treatments. For that presentation and for that work, we divided the treatments for each particular paper in two different natures of source of variability. The first one, we classified all the treatments in environmental treatments, which include, for example, different treatments of water, different treatments of nutrition, different soils, sowing dates, year, location, etc. And another main source of variation is genotypic treatments, which include, for example, different commercial cultivars, old and modern cultivars of wheat and barley, different meals, different reels. All these types of treatments were included on genotypic source of variation. Then the objective of the work was to assess the relative importance of numerical and physiological components when affected by genotypic or environmental treatments. I divided the presentation in 3 steps. The first part is related to the relationship between grain number and numerical or physiological components for environmental and genotypic treatments. The second part is different relevance of spike number and grain number per spike in wheat and barley. And the last part is differences between 2 and 6 raw power. Okay, to do that, we did an extensive revision of published paper. We revised every single paper with wheat and barley in the title, published during the last 25 years in 4 journals. We selected Field Crop Research, European Journal of Agronomy, Crop Science, and Crop and Pasture Science. Of all of these papers, we only consider data from field experiment treatments, crop simulation models, growing chambers, and pots were excluded from the analysis. To work with the database, the data were normalized to make comparison between different papers. For example, in this table, we will have the number of papers revised for wheat and for barley. In the case of wheat, we revised around 3,000 papers, and in the case of barley, we revised more or less 550. Of the total of paper revised, in the case of wheat, around 10% of the paper had results that includes grain number components, for example, spike per meter square or spike dry weight. And in the case of barley, less than 10%. And these papers, which included in our analysis. Okay, starting with the results, the third part of the results is grain number component for environmental and genetic factors. For the next 4 slides, we will have the same structure. On the left part of the slide, we will have a table with the source of variation, the environmental treatments, and the genotypic treatments. In the second column, we will have the grain number model, numerical model, or physiological model, and the variable associated, the spike number and the grain number per spike for the numerical model, and the spike dry weight and fruiting efficiency for the physiological model. on the right part of the slide, we will have the associations between the grain number response or grain number increment and the variables. In this case, spike number and the grain number per spike. In green symbols, we will have wheat, and in orange symbol, we will have barley. The first case is for the numerical component and the environmental treatments, water, nutrition, spike, so with the education years. For this, in wheat, both spike number and grain number per spike were associated with the grain number response. However, in barley, only the spike number was associated with the variations in grain number. We didn't find any association between grain number per spike and grain number in barley. Moving to the physiological components of grain number, for both, in this case, for wheat and barley, spike dry weight was associated with grain number. And there were no associations between fruiting efficiency and grain number. Moving to the genotypic treatment, and starting with the numerical components of grain number, in this case, in wheat, only the grains per spike explain the variations in grain number. And in the case of barley, the spike number always is associated with the variations in grain number. And for the last results of this first part, the physiological component of grain number and the genotypic treatments. In this case, again, in wheat, both spike dry weight and fruiting efficiency explain the variations in grain number, but in the case of barley, only the fruiting efficiency explain the variations in grain number. Okay, starting with the second part of the results, the relevance of spike number and grain number per spike in wheat and barley. Here we have a summary of the main results of the whole data set. Here we include the environmental impact, the genotypic treatments, and for wheat, for the whole data set, spike number and grain number per spike explain the variations in grain number. But in the case of barley, only the spike number was associated with grain number. To try to understand this, put the focus in one of the subcomponents of grain number, put the focus on grains per spike. The number of grains per spike is associated with the number of spikelets per spike and the number of grains per spikelets. And in these two subcomponents, there are differences between wheat and barley. For example, wheat has a determinate number of spikelets per spike because the differentiation of spikelets per spike occurs between double ridge and terminal spike. However, in barley, the number of spikelets per spike is indeterminate. Moving to the grains per spikelet, in wheat, the number of grains per spikelet is indeterminate, and it depends on the resource availability during the critical period. But in the case of barley, the number of grains per spikelets is determined because barley has just one floret per spikelet. When we, in our revision, we clearly showed that the grain number per spike variability was much lower in barley than in wheat, probably related with these differences in floral biology strongly determine for this component the determination in the number of grain per spike. Obviously, we needed to continue working, but probably this lower plasticity of spike fertility in barley could explain the higher importance of spike number in barley than in wheat. Moving to the last part of the results, and we concentrate in the differences between 2 and 6 row barley. There are 2 barley types. I think that Santiago mentioned something like that yesterday. But there are 2 barley types with differences in the reproductive outputs. 2 and 6 row barley. 6 row barley have potentially 3 times more florets per node and probably higher grain number per spike than 2 row barley. However, 2 row barley have more tillers per plant and probably higher spike number than 6 row barley. The objective of this part was to assess the relative importance of numerical and physiological components for 2 and 6 row barley. In this figure, we will have the grain number response for the numerical components, the spike number and the grain number per spike. In blue symbol is 2 row barley, and in orange symbol is 6 row barley. And in this case, for the numerical components, grain number response was only explained by spike number by both 2 and 6 row barley. Moving to the physiological components of grain number, the spike dry weight and the fluting efficiency. In this case, spike dry weight was more relevant in 2 row barley, and fluting efficiency was more relevant in 6 row barley. Okay, conclusions. For the numerical components, spike number and grain number per spike, and starting with the environmental treatments, in wheat, both spike number and grain number per spike explain the variations in the grain number. However, in barley, only the spike number is the component, is the important component. But for the genotypic treatments, in wheat, grain number per spike is important to explain the variation in grain number, and in barley, again, the spike number is the main component. In the case of physiological components, and starting with the environmental variability, spike dry weight was the main physiological component, explained the grain number in both wheat and barley. For the genotypic variation, in wheat, spike dry weight and fluting efficiency were important, but in barley, only fluting efficiency explained the variations in grain. Okay, that's all. Thanks. 18 minutes.
Excellent. Great. Roman? I have no question because I know your work.
Very solid.
Maybe you can ask Indeed someone.
I have a question. How did you collect 3,000 papers and the data from 3,000? How did you manage to do that?
6 years of work in WIT, because we start the revision
because
previous to the pandemic.
dedication was part-time, obviously. Yes.
In WIT, our dedication was part-time for all the group. And we started, I think, in 2016, probably.
Yes, you show it in your pergamino.
Yes. And well, after that, the pandemic occurs and the times delayed a lot, and we finished on 2022.
And now you can see the story in 18 minutes.
It's true. But in the case of Arg, was very different because in 3 months we did the job.
You learn how to do it.
Yes. We know the mechanism, we know the errors, and we were fully dedicated to just to
you collect data from tables and from figures?
yes, both. Tables that is easier to take, but in the figure you need to use different techniques.
I may be wrong, I don't know, but which yields are stabilized now? So, which are the best ways to increase with yields taking into account your findings?
At least for these results,
Future scenarios.
I think that the grain number in wheat will be increased by both spike number and grain number per spike, because wheat has a lot of variation or has a lot of variability in both grains. And with the same weight, the weight in the way to explain the grain number response is quite the same for the spike number and the grain number per spike. And was surprising for us because we expect some variation of spike number than grain number per spike, but the results showed that the weight is quite similar. But not is the case of barley. Barley, clearly the spike number is the main numerical component. But this probably is unexpected results. For us, I think, better results are in terms of physiological components of grain number. These figures, sorry, physiological component. This is quite new in terms of the degree of the variation in spike dry weight and fuel efficiency and the capacity to explain the grain number response.
Yeah.
There
which is the name of that? Or
water,
maybe?
conversion.
Yeah.
Maybe Jose can help in this part, but sure that water. After that, probably erosion. After that, probably soil day. And the differences between genotypes are between modern cultivars are lower. Probably if you put old and modern cultivars, the differences were higher, obviously. But I think that the water.
Good. But, sorry, current index is the same, always. The current, the conversion to grain.
Depends. For example, when we explore the genotypic variations, there are a lot of differences in current index, especially when we put or when we start looking the treatments with different old and modern cultivars.
Okay.
Here there are a lot of differences in harvest index. Probably in the other treatments the differences were lower too.
Thanks.
Considering the genetic impact, did you have a negative correlation between grain number per spike and tillers and spikes?
Both. There are a lot of trade-offs.
Okay.
I have a figure for that I didn't include in the presentation, but I put at the end. This is the trade-off between the variables. This is the grain number per spike and the spike number in wheat and in barley. This is the fruiting efficiency and spike dry weight for wheat and barley, and for the environmental and genotypic treatments. There are a lot of trade-offs. You have just only a few cases where you have positive associations by both traits. If you are in this quadrant, you can increase the brain number moving one or the another trait.
Because some new materials and Victor Sala's work are showing that wheat is reducing its ability to compete against the other plants with wheat or within the same wheat, and this could be a way to explain the increase in the number of spikes and with the same grain number per spike.
In wheat.
In wheat, yes. It reduces the tolerance, it increases the tolerance to competence, like for example. They are
early in the season. Because probably if you
I don't know. They show that they explain that some new materials, we saw something in the new materials, and the Australians have seen something that you have an increment in the spikes per square meter as a genotypic impact, and they explain that this is because there is a reducibility in the competence of the tolerate more spikes per square meter. Yes?
Okay.
So it could be something similar to what happened to Bottom.
But those cultivar has really?
Yeah, I think so. Yeah, there are the last ones released to the market. And in Argentina, we see a trend in our last paper. Alongside cultivars. A trend, not a statistical, but a trend.
A lower number of grain per spike.
No, not necessarily. No.
Probably are in this quadrant.
Yeah. This one, yeah. Yeah.
If you look the general trend,
you have a great comment.
Okay. My answer is that, of course, wheat is the main food crop for humanity. And at the end of this century, we will be above 10 million, thousand people. I suppose, I don't know. It's possible to degrade with years. Sometimes I read a paper years ago about the possibility of change the physiological machine of wheat for carbon 3 to carbon 4. Is this possible or?
maybe some
comments.
To change wheat from C3 to C4 is not a project that it's harder than it seems. I think it's the last there's a lot of developmental change that has to happen. Those are the other components. Not simple. It's very complicated.
Okay. And you don't need to increase the food production. This is not a problem with food production. It's a problem of distribution.
Yeah, sure.
Absolutely.
Yeah.
Yeah.
We need to change all our introductions in the
This is a good argument for the paper. Not the reality.
If simulations
Sorry for
said that there will be a full gap. In probably 20 simulations show that there will be a big gap in 20 years, and there is no way to increase crop yields in order to fill that gap. So there are some papers, new papers about that.
Our simulation again.
Okay, thank you very much.
We'll move to another topic. Thanks, Santiago and Daniel, for the invitation. And I'm going to talk about grain yield and his relationship with baking quality in wheat. And the relevance of the species as a crop is determined by the quality of the product, but the physiology of grain yield has received much more attention than the physiology of quality generation. And some of the variables that define baking quality are grain nitrogen concentration, as an example of a commercial quality variable, and dough baking strength has an example of industrial quality variable. And the truth is that there is a growing demand for high quality, and that implies the need for increasing both grain yield and his quality. And the need to increase wheat production to respond to the future demands is globally shared, but yield gains are frequently accompanied by a decline in grain quality. Along this change, the grains go from the field to the silos, to the mill, and finally to the bakery. The commercial quality is measured in this link of the chain, while the industrial quality is measured in this part of the chain. And according to this, with this framework in mind, our objective was to analyze the relationship between grain yield and his quality in response to phenotypic and environmental variability. For that, I'm going to focus on one particular experiment that we carry out during 2 growing seasons here, combining 3 factors: cultivars, different soil nitrogen levels, and different solar radiations incident during the grain filling period. We use 6 wheat cultivars that belong to different quality groups in which the local cultivars are classified. The first group corresponds to cultivars that are classified as improver wheats. The second group corresponds to cultivars classified that are cultivars for direct bread making. And the third group are classified as ordinary groups of cultivars. The cultivars have similar phenology and belong to different breeding companies. We use a common sowing date. The experiment was under irrigation and with control of stresses, or with dry. We take measurements in order to analyze how the grain yield was composed and how the nitrogen economy determines the nitrogen harvest. And according to this, we measure biomass at flowering and maturity, nitrogen concentration in the tissues, and industrial quality variables such as gluten, tenacity, extensibility, and strength of dough. And some of the results, I'm going to focus on the relationship between grain yield and grain nitrogen concentration, how change both traits in response to genotypes, the variations in genotypes and the environment, the physiological variables that explain yield and nitrogen concentration in the grains, and which was the relationship between nitrogen concentration in the grain and some of the industrial qualities traits that we measure. Here we have the relationship between grain nitrogen concentration and grain yield, and there is some points to highlight. The first one is that there wasn't a clear relationship between both traits. The second point to highlight is that there was great variability in both traits. Grain yield range between around 200 grams per meter square to 1,000 grams per meter square, with a mean yield of 540, while grain nitrogen concentration range between 1.5 to 2.9. The relative variability in grain yield was higher than the one observed in grain nitrogen concentration. The third point to highlight is that we can combine the high grain yield requirements of farmers with the high grain nitrogen concentration requirements of the industry in a new variable, that is the nitrogen yield that is harvested from the plot. Yeah? And these lines, the dot lines, represents combinations of grain nitrogen concentration and grain yield that allow us to harvest similar values of nitrogen yield. So the same level of nitrogen yield can be achieved through different combinations of grain yield and different combinations of grain nitrogen concentrations. For example, at these points, we have a nitrogen yield of 10 grams of nitrogen per meter square, similar to this situation with a lower yield but a higher grain nitrogen concentration. And the fourth point to highlight from this figure is that the industry demands protein concentration higher than here in Argentina, higher than 11%. This level is equivalent to a grain nitrogen concentration of 2.2. And the question is, under what conditions do we obtain proteins higher than 11%? And in our case, in 44 out of 140 cases, obtain proteins higher than the level that demands the industry. And these cases corresponds in the majority of the situations to high levels of nitrogen in the soil and situations in which the cultivars corresponds to the first group of cultivars. So a simple way to try to understand this variability is to analyze the difference between among the cultivars in grain yield, considering the response to an improvement in the environment. Here, the improvement in the environment was analyzed through the environmental index, which represents the mean yield obtained in the different situations that we have evaluated. The table summarizes the results when we have for the different cultivars. The cultivars are identified with the group that it belongs to and the name of the cultivar. And the B parameter represents the slope of this regression. Then we have the R square and the mean green yield. And the point to highlight in this figure is that the cultivars that belong to the quality group number 2, the intermediate, particularly this cultivar, presented higher yields than the remaining cultivars, the green color here. And when we have improvements in the environmental condition. And if we analyze the same results following the same methodology for grain nitrogen concentration, we have that the cultivars that belong to the quality group 1, particularly one of the cultivars, this one, row, presented higher grain nitrogen concentration than the remaining cultivars when we have improvements in the environmental conditions, the orange cultivars here. And the question now that we tried to respond was if there was a relationship between the relative response of the cultivars when we have variations in the environment in grain nitrogen concentration respect to the grain yield response. And this is the one line. And some cultivars were characterized by presenting a greater response in grain nitrogen concentration than in grain yield, while others presented an equivalent relative response, this group of cultivars. So the cultivars that belong to the first group of classification according to the quality, well, the main characteristic was that they have a greater response to variations in grain nitrogen concentration. And another point is that we try to analyze which were the main physiological variables that explain the changes in grain yield and in grain nitrogen concentration. And I'm going to focus in just one of the variables. We analyze the results through principal components analysis. And the blue corresponds to the vector of grain yield, and the red to nitrogen concentration in the grains. And this vector corresponds to the nitrogen use efficiency. That represents the grain yield obtained by each unit of nitrogen uptaken by the crop. And nitrogen use efficiency at maturity for grain yield or for producing the total above ground biomass of the crop presented a negative association with both traits. A negative association with the way in which the grain yield and grain nitrogen concentration were discriminated. And the problem is that grain nitrogen concentration has a negative relationship with nitrogen use efficiency. And the cultivars differ in the ability to establish 1 point of grain nitrogen concentration per each unit of efficiency in converting or taking nitrogen into grain yield. And this is so an alternative to obtain a balance between grain yield and the protein that we have in that grain. Yeah. For example, this cultivar was characterized by obtaining a high efficiency in converting the nitrogen in biomass, but also in higher levels of nitrogen in the grains. And nitrogen in the grains, this is the last part, is important because condition the industrial quality of the dough. Here we have the relationship between gluten and grain nitrogen concentration, the ratio between tenacity and extensibility, and dough strength. And for any variable, grain nitrogen concentration condition the baking quality of the dough with interaction in interaction with the cultivar. And here the dark shadow represents which are the levels for the different traits that the industry classify as superior. And the light shadow represents the levels of gluten or the ratio between tenacity and extensibility, and that are classified as acceptable for the industry. And the point is that grain nitrogen concentration only by itself is not an alternative for being sure about the quality of the grain. Well, taking this in mind, we can conclude that the relative variability in grain yield was wider than that explored in grain nitrogen concentration, that high grain yield and high grain nitrogen concentration were possible with high levels of nitrogen in the soil, but that imposed an economic and environmental restriction. Yeah. And managing the interaction between genotype and environment plays a key role in optimizing the relationship between both traits. And the differences among cultivars in nitrogen use efficiency, in nitrogen partitioning to the grains, as well as the analysis of allelic partner for protein composition constitutes the way to analyze this interaction. Well, this is our working team that was behind these results. Thank you.
I don't remember the management of nitrogen in terms of How many stages?
We work with 3 levels of fertilization, with 3 levels of nitrogen in the soil. Yeah, that we obtain the levels through fertilization, and considering also the native soil at sowing. And the levels were 60 kilograms of nitrogen per hectare, 180 and 240.
The fertilization was?
At sowing. Not at 50 days after emergency.
Okay. Do you think that the results will be changed if you modify the fertilization strategy? For example, fertilization in tillering or doing standard irrigation or directly to the flag leaf? I mean that
yeah.
Which is the weight of the fertilization management in this type of result?
There is here in our results a point of attention, because our experiment was under irrigation, but under field conditions, I haven't expected a big difference with partitioning or not the nitrogen, unless that the fertilization were late in the cycle.
I think there was like kind of a tendency a few years back where if you fertilize a foliar fertilizer in flat leaf, you could get to 1 or 2 points of clothing extra, and maybe that could help in the efficiency.
Yeah, sure. For that,
it's just specialty, like it's not a regular management, so it's kind of costly, so it will be a
just with late fertilization, it's possible to obtain changes in the protein, but it depends also on the basal level of protein. If we have high levels of protein, about 10%, the possibility to increase the level of protein is reduced.
So that was a lovely talk, very interesting. I'm really interested in the relationships between grain yield and growing nitrogen factors that can influence that. And I saw that you have no relationship between grain nitrogen content and yield, whereas actually you would expect an inverse relationship, right? Which you did see when you calculated the sort of yield you used anyway. So I thought that was lovely, but I thought that maybe the reason you see no relationship, it's not, well, yeah, it's the stage, I guess, between grain nitrogen content and yield, is because you've got those 3 big different nitrogen treatments. And what you really need to look at with this plot is that those 3 concentrations separately.
We have here the relationship for the whole data set. Yeah? Yeah. But if we analyze the data by cultivars, we obtain, well, I have it here.
I'd like to see the data analyzed by nitrogen
treatments.
No.
The nitrogen treatments.
Yeah. If we analyze the data, separating the genotypes by one side and the nitrogen in another side, we have a negative relationship. Yeah. So we analyze not for the full data, but I have, they are very small, the table here, I think. I'm not sure if I have here. Just give me a minute. No, I haven't here the figure. No, I haven't here. But for this is very nice information. Nitrogen. this is culture. And if you analyze individually, each factor will have a negative correlation. But when we compose all the data sets, this negative correlation changes fascinating.
Yeah, it doesn't surprise me. But the fact that you saw a lovely negative relationship when you plotted against anyway, I thought that was really nice. But
That's why take relevance the concept of interaction between
nitrogen supply, soil nitrogen is going to influence yield and dependent but relating. That's good. Anyway, thank you.
Okay. Thank you very much. We are tired. We try to make a fast talk, if I can, briefly. Where is my talk?
Chica. Mirales.
No. Ahí está.
Aquí.
Ahora. Ok.
Aquí.
Hay que compartir la pantalla.
Te compartimos.
Duplica. Ahí. Okay. I'm going to talk about the diseases. Okay. I can eliminate that. Okay. Huh?
Go to with the mouse.
Okay. I'm going to talk about the effect of the diseases on yield and the commercial and industrial quality in wheat. During these 2 days, we mentioned the different yield potential in different environments, and diseases appear in this sector of the rechargeable yield between this yield and the potential. This is one of the reduction factors, which is one of the most important reduction factors of yield and quality. And where the diseases affect the yield component from the physiological and numerical components in cereals. Normally, if we have this sequence from the incident radiation to yield passing through by the mass, one of the main effects of the diseases is on the leaf area index, reducing the leaf area index. This is the relation between intersector radiation and leaf area index. And the diseases appear crazy, the laser point. The diseases reduce the leaf area index and also reduce the intercept radiation. And the other point is that different diseases not only can reduce the intercept radiation, like in these photos, because you have a necrotrophic diseases like tan spot or biotrophic diseases like leaf rust. When you put the radiometer below the leaves, the radiation interception is the same in this diseased leaf or affected leaf and in healthy leaves. And this is the reason that we create a new case. This is the absorbed radiation. This trait is trying to determine which level of radiation that is intercepted by the leaf really is absorbed by the crop. Because in general, the normal sequence is incident, intercept, and biomass. but in diseases it's necessary to include this box, the new box, the absorbed radiation. And this is what happened. With the same leaf area index, this is the intercept radiation, but in real, depending on the effect of the diseases, you can obtain a very different absorbed radiation. And this is some examples of different diseases that affect intercept and absorbed radiation through the leaf area index. And this is, again, what happened with the diseases that not only reduce the leaf area index and obviously the intercept radiation, also can reduce the absorbed radiation. The other important trait is the radiation use efficiency, because some diseases not only affect the intercept and absorbed radiation, can also affect the radiation use efficiency. And this is an example that, or a speculation that, if you have this line between biomass and absorbed radiation in a healthy crop, this is what would be happen when you have a diseases that could be affect the radiation use efficiency. I make a compilation of different papers that we published related with diseases. Most of these papers were based on field experiments like this here in the faculty, where we infect the crop with a combination, a cocktail of different spores of different rust diseases. We concentrate mostly in rust, but we will know that at the field you can induce the rust, but appear other diseases that are in the straw and in the field. But we try to push or put the emphasis in the rust, stem rust, leaf rust. We apply the spores and we cover the crop with these tents during the night only for a couple of days to promote the infections. Mostly during 3 or 4 days was enough to promote the diseases. And most of the inoculation was applied during the critical period or previous during the critical period to affect the critical period, or sometimes during the grain filling. It's not easy to work with diseases because it's different to put a shaded or cut the leaves, or where you are exactly know when you apply the treatment. In this case, depend the weather, depend the different things. But we obtain this. This is the leaf rust, an infected crop with leaf rust. And also we obtain in other crops infection with stem rust. Take account that the stem rust first of all appear in the leaves and after that continue in the stems. Okay, some results. This is the area and the diseases curve progress. This is a common quantification of the diseases in the crops. We contrasted the yield against the area under the curves, and we see in this figure, this is the healthy crops here, and this is the diseased crops. This is in absolute values, and this is in relative values respect to the mean of the whole experiments. And at the same time, we observe reductions in the grain weight when the diseases appear during the grain filling and reduce the leaf area index. The grain weight was reduced in the diseases crop respect to the healthy crops. And we also measure other traits that were related with the biomass accumulation. For example, you can see in this figure the green leaf area index against the degree days for ananthesis during the grain filling in different years. And it's interesting to see here, this is the leaf area index of the crop that was protected, the leaf area index, the green leaf area index, in the healthy and in the diseases crop. What is interesting to see here, this line represents the critical leaf area index. Depend the year, the diseases, the diseases crop remain for a couple of days or weeks above the critical leaf area index, but after that, the diseases crop was below during most part of the grain filling, below the critical leaf area index. In some cases, the control remains all the time above the critical leaf area index. In other cases, both diseases and healthy crops were below the critical leaf area index, and so. And we have different combinations. And it's interesting to combine this with this because we can see here that, for example, in this case, where the crop remained, the healthy crop remained all the time above the critical leaf area index, there was not important reduction in the radiation interception. But once the crop the diseases crop start below the critical leaf area index, the intercepted radiation was reduced. And we can see this example, for example, that at the beginning of the grain filling, the crop was a very low value of leaf area index. The crop was fastly, or in a couple of days after anthesis, started to reduce the radiation interception below the 60% of the capacity. And in other words, depend the leaf area index that was affected by diseases can impact in different manner with the radiation interception. And for example, in this case, it is clear that the green area index, when the diseases attack in very important manner the leaves, the leaf area index immediately was below the critical, and the radiation interception dropped a lot, very sharp. The other point is we measure, or we're trying to understand what happened with the photosynthesis in the leaves. This is healthy leaves, and this is leaves affected by leaf rust. And when we show this figure, we always ask the same question: is how much is affected these leaves by the rust? Do you know? Which proportion of the leaves is affected by leaf rust? 30%. The rest?
That's probably more.
40.
50.
50.
For me, it's less. 25.
25. It's very interesting because if you apply digital images on this image, you can exactly know which is the proportion of the leaves that is affected with rust. And the people that go to the field to observe the damage, normally when the leaf is affected in a low proportion, the human eyes probably overestimate the damage. And when you go to the field and you have a lot of an important damage in the leaves, is the reverse. You normally underestimate the damage. When we apply digital images, this is 20% the damage. 20%. Yeah. And now what we did is correct. We measure the relative photosynthesis in the healthy leaves, and we measure the severity. This is the severity, 20% of the severity in the leaves, and correct the photosynthesis with the severity. 80% of the leaves in theory is healthy, and if we measure the photosynthesis in this leaf and this leaf, not only leaves, in a wide range of severity, if you observe a damage of 20%, your photosynthesis is 80% in these leaves. And this is a one-to-one relationship in theory. But when we measure the photosynthesis in a wide range of severity, we observe this. Photosynthesis, when severity increases leaves, the proportion of reduction in photosynthesis is higher than the severity that you observe in the leaves. In other words, this, in theory, a healthy part of the leaves is not produce the same photosynthesis as the same part of the same proportion in the healthy leaves. There are some part of these leaves that even when you see in the field, in the crop green, is affected by the diseases. And this is leaf rust. When we apply different nitrogen in the crops, and you can see that there are a combination, but most concentration of the points are here with less severity than here, that we observe more severity, especially with more nitrogen. The more nitrogen you apply in the field, in the crops, more leaf rust severity you observe in general. There are another diseases in the field that appear because they're in the straw or in other crops. And it's interesting when we try to separate leaf rust and septoria, all these points with less nitrogen correspond to septoria. And all these points with high nitrogen mostly were leaf rust. The prevalence was higher in leaf rust here than in here. The conclusion with this graph is that if you have some necrotrophic diseases, septoria is not exactly a necrotrophic disease, it's a mix between biotrophic and necrotrophic, but in general, if you have necrotrophic diseases, you have a correlation with a one-to-one relationship. But when you have other diseases, for example, biotrophic like rust, most of the points are below to the one-to-one relationship. This is septoria and this is rust. The same experiments, we did the same experiment in Grignon. Roman spent time there in France in Grignon and measured the same approach, and we observed exactly the same in Grignon than in Buenos Aires. That was the experiment that I mentioned before. The other interesting point that we want to know is the question that when you have rust in the leaves, affect the temperature of the leaves or affect the VPD, the VPD, because you know that the rust destroy the epidermis and probably the water can be lost from the leaves more than in healthy leaves. Our results show that diseases did not affect leaf temperature because it is a one-to-one relationship. But the VPD in general, when we have rust, we observe that the VPD was slightly increased here respect to the healthy leaves. And in other words, the diseases affect the BPD, but did not affect the temperature. And the other approach was related with the how much resource you have to compensate the grain weight when you have the leaves in the crops. Because if you have not a green area to produce carbohydrates or to produce to obtain photosynthesis, probably the grain weight is affected, especially if you produce a lot of number of grain per meter square. And we observe in this graph, we have the relative change in grain weight and the healthy green area in the healthy green area here. And this was measured. This is the megajoules per grain per milligram, or the megajoules per grain. This is correct by the grain weight. This is not correct by the grain weight. How was the capacity of the crop to intercept this radiation per number of grain produced? This is this axis. And we observe that as the relative change in grain weight is decreased as you can capture more radiation during the grain filling per grain that you change in the crop. Until this, there are a threshold here, and if you are to below the threshold, you increase the relative change in the grain weight. And this is also in part related with the the water soluble carbohydrates that is represented in this axis, and this is the days fermentation during grain feeding. This is the dynamic of carbohydrate in a healthy crops, and this is an example of the dynamic of carbohydrate in the diseased crop. We observe that in the healthy crops, you can accumulate more carbohydrate in the peak, more than in the diseased crop, but at the same time, the diseased crop use the carbohydrates early than the healthy crops, and consume, in this case, not more faster because the slopes are the same, but in other cases, we observe that the consume of this carbohydrate to compensate the lack of photosynthesis is more faster than in the healthy crops. In summary, in the healthy crops, during the grain feeding, most of the sugars come from leaf photosynthesis. Next, sometimes can use the reserves, and finally, the spike photosynthesis can increase. In some cases, that is not enough. In which is not enough the leaf photosynthesis and the reserves. But in a diseased crop, the leaf photosynthesis is affected. The reserve is very important because use most of the reserve in the stems mainly, and the spike photosynthesis is demonstrated. Roman works in Spain with this approach and demonstrate that if you have not enough leaf photosynthesis and the reserve are not enough to compensate the grain weight, the crop increase the spike photosynthesis and also the peduncle photosynthesis. The last part of this talking is related with the quality. We measure different traits of the industrial quality. This is a representation of the alvegram. This is the tenacity, the extensibility, and the Dow strength. This is the integral of the area under the curve. And this is a tedious table, but I try to resume this. This is the control, the diseases in different sowing dates. We use different sowing dates because, in general, when we apply stem rust appear in the later sowing date in general, more than in the early sowing dates. But the most important effects are these. This is septoria and this is stem rust. Septoria is a necrotrophic diseases or mix between necrotrophic and biotrophic, and modify some of the parameters. Grain protein, reduce the grain protein. Remember that in this case we have a control with fungicides applied all the time, and a disease that when applied the rust, we didn't apply any fungicides. We didn't correct the attack of the rust or the septoria in this case. And in both cases, septoria reduced in both sowing dates the grain protein concentration, the gluten, reduced the gluten, the dry and the wet gluten was reduced, especially in these sowing dates. And when the stem rust is present in the crop, especially in the late sowing date, this is the late sowing date, the reduction of the parameters is dramatic because reduce most of these parameters, especially the nitrogen grain concentration, grain protein concentration, this is the grain protein, and also reduce the wet and dry gluten in both years. And the other result is that when you have a diseased crop, even with the same yield, if you have the diseases, the grain protein concentration is reduced with respect to the healthy crop. It's not only the problem with the yield, the problem is also with the same yield, you have less protein. This is the result of the wet gluten. This is the relationship between wet gluten and protein. Gabriela shows a very close relationship. There is, in general, a very close relationship, but you can see the diseases reduce wet gluten and grain protein in the same way, and also reduce different other industrial parameters. For example, the drought strength is reduced in the diseased crop respect to the healthy, the cell intensity reduced, and also grain protein. Drought strength is mostly related with gluten. This is a very fast test to determine industrial quality, which has less correlation, but the diseases again is reduce these parameters and also reduce grain protein. Okay, some conclusion. Both diseases, necrotrophic and biotrophic, penalize yield, but in different magnitude. It's worse in biotrophic diseases if you don't control these diseases. And most of the diseases from heavy and watering the grain filling, affecting grain weight, and in some cases can affect grain number because can abort in the last part of the grain setting, can reduce the grain setting if diseases appear very close to the anthesis. There was a positive relationship between grain weight and green leaf area index in the diseased crop, and the necrotrophic and biotrophic diseases reduce intercept and absorb radiation, both by biotrophic diseases. And also, in some diseases like rust, reduce the photosynthesis of the apparently healthy leaves in the diseased leaves. When crops are infected by diseases, the reserves are accumulated in less magnitude and in some cases are used more fast. Grain protein and gluten are reduced, and although both necro and biotrophic diseases reduce most of the industrial parameters, baking and millet parameters, the penalization in all cases was higher in biotrophic than in necrotrophic. And this is the diseases group
Thanks.
Yeah. Okay, thank you. Questions?
Well, it's more of a methodological questions. Because there's a lot of fungi that have aerobic respiration. So if you're measuring photosynthesis and you have rust, probably rust are also releasing carbon dioxide when you're measuring photosynthesis. That can probably have some effect on the way you measure photosynthesis.
Yeah.
I don't know if rust have aerobic respiration, but many fungi have that kind of respiration.
Yeah. When this is one of the black box that you have into the system when you measure that. Yeah, that's right. Yeah. It is true. Yeah.
We measure. Sorry, can I?
Yeah, sure.
We measure during the night. And in these experiments, the increase in respiration during the night with the same methodology was.
Light.
Yes. I don't remember exactly the numbers because Ramiro is involved.
Yeah, Ramiro measured it.
More than me. But the rust have aerobic respiration, the fungi, rust.
Yeah.
Very useful. It's not significant.
It's true that you mentioned, but when we measure during the night, the increase the log is not significant, but it's not right.
Yep.
Funny. And probably for Mansu or Ramiro, but how did you set with the software, which was the exactly color change, the threshold to determine that this was effective or not? Because there was some
As I remember, we use software. I don't remember. Do you remember the name?
Yes, it's a software from the American Phytopathology Society.
Yeah. I don't remember the name exactly. The software allows you change the precision of different color combinations.
Yeah, I remember. I used it for top color.
Yeah.
It was easy. It was brown and green.
We try to apply the more realistic combination of the color to capture the rust, the spore of the rust, and the threshold of the yellowish of this halo that is around the spore. But if you change the criteria, obviously change the area. Yeah. I don't know, Romani.
No, all the time we move the threshold. Imagine, to try to capture the more realistic situation. But for example, Colin Robert has a very nice paper demonstrating that the dimension change a lot the results in terms of photosynthesis and severity. Because if you include, for example, only the spores, you could find some type of results. But if you include more areas, for example, yellow areas in the measure of severity, the results will change a lot.
But still, there is some damaged tissue that you are not seeing.
Yes.
And the fungi is there.
Yes. But the quantity of this part depends on the image analysis. Because if you include more or less area in the x-axis.
Yeah, but the trend is the same.
If you're just using an Arg-Uk, probably just not going to see that. If you could get hyperspectral information, you would be able to see areas of information that are not visible.
Yeah.
Because they'll be doing things to the other properties of the practically in the technology.
Yeah, this is policy with Arg-Uk.
Yeah. Or if you could image it with fluorescence, you'll be able to see what.
I'm not at all surprised that the impact of the infection goes beyond where the chlorophyll has been lost. When you ask what was said,
I'm sure that was
this is my answer. What I said, just saying.
First, really interesting the approach for the management of PFAS. But from 2017, we have yellow rust as the main disease in Argentina, and this begin or start early in the cycle, many times from Thailand. Do you believe? I imagine that the impact will be greater, but do you believe that the approach could be useful for the management in general, or you need to adjust?
Two things. Obviously, in all the crop, or in most of the crops, exists what we call the critical period. The critical period is clearly source limited. In all the crops, when you identify the critical period, it's a critical period because you have a lack of source to produce seed. If yellow rust appear in this period, I'm pretty sure that the impact is quite higher than if appears during the field, because you affect especially the grain number in the square, and maybe you cannot affect the grain weight, depend the reduction in the grain number. If you apply a big reduction in the grain number, the grain weight can be a little bit compensated, but or maintaining the same level. This is the effect on the yield. But the physiological effect, when you compare the yellow or the orange, the rust, for me, it's exactly the same. Yeah, because the problem with the rust is that it infects the intercellular spaces, and this is affect the photosynthesis and other attributes of the leaves. But for me, both rust, I don't know, because we didn't expect yellow rust in this, but yeah, for me the effect is the same. From the physiological point of view, it would be the same.
If maybe yellow rust affect more the number of pillars because it's very.
If appear in that moment, probably, yeah. But if appear during the grain feeding, it's the same than the grass. Yeah.
And this approach is just in 2 dimensions, because the leaf area index is just the sum of the total area of the leaves. But in fact, the canopies are 3 over dimension. And if you put this another dimension, probably you have differences in terms of the stages which the grass affect. Because it's not the same during the critical period, because you have a lot of leaves in their maximum leaf area index that in the beginning of the stage.
So I'm going to give the last talk. I'm going to try as fast as I can. I have just 100 slides, but people fast. Don't worry. So I'm going to really change the topic. I'm not going to talk about crops, but I'm going to talk about plants that compete with the crops. And my name is Eduardo, and first I want to thank Daniel and the organization and all the people that organized this workshop for inviting me. And we have been working on the last 20 years trying to understand how the environment regulates wheat seed bank dynamics. And in this talk, I'm going to show how we understand how seed banks, the dynamics of the seed banks, of the wheat seed banks, and how we can use that to develop a model in order to predict the risk of wheat emergence in the field. So I'm going to divide the talk in 3 parts. In the first part, I'm going to show what is the utility of predicting the immersion of weeds in the field. And then I'm going to present a framework for understanding how seed bank emergence patterns are regulated by the environment. And in the third part,
I'm going to show how this framework can be used to develop a simulation model to predict the risk of weed immersions in the field. So I'm going to start trying to answer this question: what is the utility of predicting the immersion of weeds in the field? And in relation to this, we can say that the success of weed control methods depends largely on the possibility of reaching the greatest numbers of weeds in the seedling state, because in that state, plants are more vulnerable to control practices. We all know that if plants are smaller, they are easier to control. And in this slide, we have the optimum size of two very important weeds, horseweed, Conisa, and pigweed, Amaranthus, is the optimum size for the application of post-immersion herbicides. Okay? So you can clearly see that plants, in order to get a good control, plants have to be very small, probably 1 week after immersions. And probably one of the main problems, or the reasons why we don't have very efficient weed controls, is because farmers usually try to control weeds when they are larger than the optimal size. And we can see this here. This is a survey of farmer consortium called APRESID, that many of you know. And in this survey, they ask different farmers from the different zones of Argentina, the nuclear zone, central and south and west zone, which were the main limitations that affect weed immersions. And most of the farmers say that the main limitations were late applications with weeds larger than recommended. So it's clear that if we can predict the moment of weed immersions, it can be useful for designing more effective control strategies, basically because we are going to be able to control weeds when they are small, with the optimal size for controlling weeds. I'm going to illustrate this with this slide. So if we are able to predict when weeds are going to immersions begin and when immersions ends of a certain weed cohort, we, for example, can know when is the optimal moment for the application of post-immersion controls in order to affect a higher fraction of the weed population in the seedling state, but also when we have to apply pre-immersion control herbicides and how long this control should last in order to affect a higher fraction of the weeds during the immersions. Okay? So if we can predict seedling immersions, this can be used to optimize the moment of the application of control practices, also to increase the environmental efficiency of controls, because we can control weeds with a lower dose of herbicides and with a lower number of applications, and also to scatter weed scouting in the field, because we have some type of alarm that is telling us when a certain weed is going to emerge in that plot. We can go there to scout the weeds and to control the weeds with a good timing when they are small. Okay? So, what processes do we need to know to predict the immersion of weeds in the field? And if you see this slide, you are probably thinking about germination. And well, you are right. One of the processes we need to know is germination. As you may know, germination is basically regulated by two factors, temperature and water. And we can predict germination in relation to these two factors, basically using threshold values, like the minimum temperature or base temperature for germination and a minimum soil water content for germination. So when soil temperature is above the base temperature for germination and soil water content is above this minimum soil water content, we can predict that seeds are going to germinate and emerge. Okay? But is this enough in order to predict the emergence of weeds in the from the seed bank in the field? And unfortunately, this is not enough, because seeds of many important weed species present dormancy. Roberto talked about dormancy yesterday, but in order to remember what is dormancy, basically dormancy is a blocked germination of a viable seed under favorable conditions for germination. This means that in order to germinate, these seeds have to be unblocked and dormancy have to be lost before seeds can germinate and emerge in the field. So why is important to take into account dormancy if we want to predict weed immersions? Because basically the timing and extent of weed immersions is strictly related to the dormancy state of the seed bank. And so for weed seeds that present dormancy, taking into account changes in seed bank dormancy level is essential if you want to predict weed immersions patterns in the field. And you can see that here I'm talking about dormancy level, not about dormancy or no dormancies. That's basically because dormancy is not an all or nothing phenomenon. You don't have just dormant seeds or non-dormant seeds. You can have seeds with different levels of dormancy. And this is represented by these emojis here. So you can have seeds that are very dormant or almost no dormant, and between these two states, you can have different levels of dormancy. And how these dormancy levels are expressed, or what means that seeds have different dormancy levels. And in relation to this, we can say that the level of dormancy establishes the range of temperature in which seeds are able to germinate. So, for example, seeds that have a high dormancy level, they can germinate in a very narrow range of temperatures. And as dormancy level decreases, seeds with a low dormancy level can germinate in a very wide range of temperatures. And so when the dormancy level decreases, there is a widening of the range, and when the dormancy level increases, there is a narrowing of this range. In many relevant agricultural weeds that usually emerge in spring or late winter or early spring, that we call summer annual weeds, they probably emerge in late winter, early spring. They grow and develop during the spring and the summer, and they disperse the seeds in in the beginning of the autumn or in late summer. These changes in the range of temperature in which seeds are able to germinate are a consequence of changes in the minimum temperature of germination. So as seeds lose dormancy, they are able to germinate at lower temperatures, and as dormancy level increase, they lose the capacity to germinate at low temperatures. Some important thing about these changes in dormancy level, when I'm talking about these seeds, I'm talking about seeds in the seed bank, in the soil, okay? That they present, with seed bank, show seasonal changes in the dormancy level. So seeds in the soil are changing the dormancy level in a seasonal way. And I'm going to show this in this diagram. Here we have the seasonal changes in seed bank dormancy level for a summer annual weed. And this is the seed bank dormancy level and the different seasons of the year. And usually seeds are dispersed in autumn with a very high dormancy level. This dormancy level decreases during winter, they have a very low dormancy level in spring, and then this dormancy level increases again during summer, and we got again a high dormancy level in autumn. And in the other year we have exactly the same pattern. So these cyclic seasonal patterns of changes in seed bank dormancy level in summer annual weeds are regulated by soil temperature. And in these weeds, in the summer annual weeds, cold temperatures during winter provoke dormancy release, while warm temperatures during summer provoke dormancy induction. So in this way, soil temperature regulates the seasonal variation in the dormancy level. As I said before, this variation in seed bank dormancy level are related to changes in the range of temperatures in which seeds are able to germinate. So we can plot that here, and for summer annual weeds, this range of temperatures in which seeds are able to germinate is here in something like yellow, is limited by a higher limit temperature that doesn't change, which changes in dormancy level, and a lower limit temperature. This is the temperature that changes according to changes in seed dormancy level. So how this works for the seed bank? In summer annual weeds, cold temperatures provoke a decrease in the dormancy level, which is related to a widening of the range as a consequence of a decrease in the lower limit temperature. Summer temperatures provoke an increase in seed dormancy level, which is associated to a narrowing of the range due to an increase in the lower limit temperature. So, When immersions take place in the field, well, when the soil temperature overlap the range of temperature in which seeds are able to germinate, and this usually takes place in spring, where rising temperatures surpass the lower limit temperature. So then here's in an overlap between the thermal range under which seeds can germinate and soil temperature, and that provokes immersions. Then when it stops, well, basically when temperature goes out of range. So if we see this slide here, we can clearly see that if we are able to predict the relationship between soil temperature and how this lower limit temperature change, we can be able to predict when there's going to be an overlap between soil temperature and the thermal range for germination and when immersion is going to take place. And basically, that's the way the model works, okay? I'm not going to show the equations because the model has something like 40 equations, but the model simulates this, okay? Make a simulation of this, establishing a relationship between the soil temperature and how this parameter here, this lower limit temperature change. But the model also have other factors that can affect wheat germination in the field. Maybe you probably know that some wheats require light to germinate or alternating temperatures to germinate. So once temperature is within the range, many wheats are stimulated by what we call the dormancy terminating factors. And in the model, we include two dormancy terminating factors, light and alternating temperature. These factors basically removes the last impediment for seed germination, stimulating germination. Okay. Why they are important? Because they have very important ecological meaning in the field. So that's why many wheats require these factors. So these two factors allow weeds to detect the right place for germination. So if you have soil cultivation of canopy thinning, there will be an increase in the red fiber ratio, and this will promote dormancy termination through the phytochrome system, and also a wide daily thermal amplitude. So dormancy is going to be terminated, and the weed seeds are going to germinate. But if the place is not good for immersions, for example, if seeds are too deep in the soil, there will be no light, a very narrow daily thermal amplitude, and dormancy is not going to be germinated. Or if seeds are below a very dense canopy, there will be a very low red fiber ratio, thermal amplitude, and dormancy is not going to be terminated. So there is a very important ecological implication of these two factors. So based in this understanding of the system, we developed this conceptual model when we have we developed some equations that can predict how this lower limit temperature change according to soil temperature. And then we can predict when temperature enters the range, and this provokes immersions. I showed that before. One of the equations of the model is related to this phenomenon. Then we have another equations that they take into account the effect of alternating temperatures and light on seed germination, on dormancy termination. And then we have some equations that predict the effect of temperature and soil water content on the germination process. So basically, this is the conceptual model we use to develop the model. Okay, I say I'm not going to show all the equations, but this is the conceptual model. Okay? We just put equations to make this work. And the model also takes into account the tillage system, because this is very important, because the tillage system can, if you are no-till or you are conventional tillage, this will change the position of seeds in the soil and which temperature they experience, which soil water content they experience. The stable volume is the salt on the soil, that also this will affect the soil temperature and soil water content. And also the degree buried seeds are exposed to light, because if you remove the soil, a higher fraction of the seed bank is going to be exposed to light, and if you don't remove the soil, a lower fraction of the seed bank is going to be exposed to light.
And this is basic functioning of the model. But one important thing is that our model does not predict seedling emergence, how many seedlings emerge, or how many weeds emerge, but we predict the risk of emergence. Okay? And this is a different concept. This risk of immersion can have values between 1 and 0. And I'm going to show now how you can you calculate the risk. Basically, this risk of immersion can have values between 0 and 1, measure the degree to which the model conditions for immersions are met. Okay? So for each of these processes, say dormancy level regulation, dormancy termination, and termination, you can have different values between 0 and 1, and you have to multiply those values. So if the conditions are optimum for seedling immersions, you got 1 for this process, 1 for this process, and 1 for this process, and you have the maximum risk. If some of these conditions are met partially, you have a value between 0 and 1, and the risk of immersions is between 0 and 1. And if one of these processes doesn't meet the condition, for example, you have water in the soil, you have a value of 0, and the risk is 0. Basically, it's very simple. So, which are the inputs of the model? Well, we have the soil type, some weather data, soil temperature between 0 and 10 centimeters, air temperature and rains, some crop management data. We have to know the predecessor crop and the yield in order to take into account the effect of stubble on soil temperature and soil water content, and also the tillage system. And with that data, the model used the following drivers. Soil temperature, that is the same as the input. Soil water potential, because the model have an internal equation, some equations, a routine that with the rains and the soil type can calculate soil water potential. And then the daily soil thermal amplitude to calculate alternating temperatures, and the degree seeds are exposed to light is related to the tillage system. If you are in a conventional tillage system and not a direct drill, this is related to which is the degree of exposition to light of the seeds. So this model is available on the web, and it can be used by technicians or producers. And this is the website. And the user have to put the weed, the location, the tillage system, the predecessor crop, and the crop yield. And with the location, the models get the soil type and make an automatic capture of weather data. The average daily soil temperature, rainfall to predict soil water potential, and daily thermal amplitude. But one of the interesting things of the model is that when you consult the model, the model get an automatic capture of weather data, not just for the day you consult the model, but the model used a 7-day weather forecast. Okay, so with that 7-day weather forecast, we run the model, and so what you got is a forecast with immersion risk model, because you can predict which is going to be the risk of immersion of that week for the next 7 days. Okay, so you have a daily immersion risk forecast in the next 7 days, and this is basically like a climate forecast. Instead of having if it's going to be sunny, heavy rain, or light rains, the model predicts if you have the risk of immersion is 0, you can have a risk between 0 and 1, or you can have a very important immersion of the weed, okay, or you have a higher risk of immersions. That is how the model works. I'm going to show now the website. So the model is called PRORIEMA, okay? Spanish is Pronóstico de Riesgo de Emergencia de Malezas. It's like the forecast of the risk of weed immersions, and you can consult we have different weeds. We have Echinoclacus galli, Amaranthus hybridus, Polygonum avicularis. This is a very important weed of wheat crops. and these are more summer crops. And we also are developing some for logging for ryegrass at the moment. So the user have to put the weed, the location, the tillage system, the previous crop, and the previous crop yield. And with that information, the output of the model is this. You got a clock that you have, which is going to be the risk of immersion from the next week. This is from the 23rd of September in Balcarce, in the south of Buenos Aires, with conventional tillage sunflower that yield between 3,000 and 4,000. And for that time, the risk of immersion was low. And then also the model gives you the evolution of the risk of immersions from the beginning of the year to the day you consult the model. And we validate this model in a lot of places of Argentina, basically in the Buenos Aires province. And I'm going to show now the validations of the model. This is a validation of the model in a wheat crop in Barro in the south of Buenos Aires, Argentina. Here you have how the model predicts the decrease in the lower limit temperature and the increase of the lower limit temperature. This is soil temperature. And the model predicts the immersion of weeds when soil temperature is within the thermal range permissible germination. And here you have the prediction of the model in green and seedling immersions in red. And you can see that the model makes an acceptable prediction of the pattern of weed immersions. This is also a validation with data some researchers from Spain sent us. This is wheat under mold law in Spain, LEIDA. And here you have how the model simulates changes in the lower limit temperature when the temperature overlaps the range. This is the soil temperature is this blue line. And this is the simulation of the model, which is the one in green, and they observe seedling immersions in the field. And this is the same, but for a dry year. Okay? It's another year in Spain, but it was really dry, and the model was able to capture the defect of low water potential and defect on seedling immersions that decreased the risk of seedling immersions. This is some validation for Amaranthus hybridus. Here you have the simulation by the model of the low limit temperature and the soil temperature. And here you have the risk of immersions predicted by the model, the green one, and the seedling observed in the field. And here we have two very important dry spells. This is soil water potential. And we can clearly see that the model gave an acceptable prediction of the pattern of immersions and also was able to take into account this very important dry spell during the immersion season. And finally, I'm going to show these are some recent modeling approaches we are using for ryegrass because we are developing the model for this weed. And this is for 2 years in Pergamino for ryegrass, 2019 and 2018. And you have the seedlings observed in the field. They are in yellow and the prediction by the model in blue. One important thing here is that in 2019, the weed emerged earlier, and the model was able to capture these earlier immersions if you compare that with 2018. So, in order to finish, how you can use this information, or I want to show just a potential practical application of the model. This is for ryegrass. If you have, this is the prediction by the model, the blue one, the risk of immersions. And for example, you plant a wheat crop. This is in the southern hemisphere in winter here in June. Okay. If you have this information by the model, you can plan different tactics, the control you follow, and you can use, for example, grass-based herbicide, residual herbicide, or probably no chemical control. You can use the predictions also to plan a strategy to include a pre-sowing or pre-immersion herbicide. These are two herbicides that are used in wheat to control ryegrass. And finally, if the model is you have a prediction of immersions, during the post-immersions of the crop, you can also plan to apply other type of herbicides. So, as conclusions, we can say that based on a conceptual framework of how environmental and management factors regulate the functioning of wheat seed banks, we develop a model that allows predicting the risk of wheat emergence in the next 7 days. So it's a forecast. This model is a tool available online and connected to climate forecast. The model was validated against field wheat emergence data, showing an acceptable prediction of the risk of wheat emergence. And also the model has a generic and mechanistic character that allows to reuse the model for more than one species. If you have the equations, you can reparameterize the equation for different species. It also allowed an understanding of the processes involved in the regulation of wheat emergence and have the possibility of a continuous improvement of the model, because you can test the model against seedling emergence data from the field, and you can try to adjust the model to get a better prediction. So, two people that are involved in this work is Diego Ferrado, that works with us in the crop production, in the crop production institute, department, and he's an expert on models, basically. And Christian Malaver, that is working on weeds and is part of the team. So thanks for.
Thank you.
I know probably it's going to be very complicated. It's a very complicated thing to incorporate, but you don't have an input to the model saying what was the pressure of that weed in a specific field previous seasons. Is there a way that, or did you consider including some type of information of that? I'm thinking of like predicting an emergence threshold above which you can expect large yield increases in the crop next season.
But you can include that. We didn't do it, but you can include that. One problem with the model is why the model predicts a risk and not the number of seedlings. That's because no one knows how many seeds you have in the sub-seedlings. So there is no point on predicting a fraction or there is no point in predicting how many seeds are going to make, how many seedlings. So if you know that in your field plot you have a problem, for example with amaranthus, or you have a weed that is a problem, you have to check that away. But probably the model is saying, okay, you have a very important risk of emergence of Conisa, but you don't have Conisa. Conisa is not going to emerge. So the model is just saying that this is a risk. Okay. And if you know which is the important weed, the problematic weed in your plot, you can look for that weed. One thing we're trying to do is we are mixing this model with, ProReapers. It's a model that calculates which is the best combination of herbicides to get a more sustainable application or lower toxicity. So we're planning to mix both models in order to make a recommendation of which herbicide you can use to control those weeds. That's what we're planning. And the only problem with the model is that you can just predict 1 week. And it's very short. But the problem is that when we talk with all the people from the National Weather Service, for the service, they told us that more than a week is just. It's not real. But it would be great to have a longer time.
Yeah, of course, because you have the power to predict if you should wait more or apply.
And the model has
which species are arriving?
Which species? Now we have Epinophilus galli, a pin is very important summer weed, Amaranthus civilus, that is pigweed in English. We have Polygonum angular proselynoid, is a very important weed of crop, weed of wheat. And we are developing ryegrass, because ryegrass behaves the other way around because ryegrass emerged in autumn and not in spring, not in winter, so or basically in autumn. So the way we develop the model is different. And we are developing the model for ryegrass. And what we are trying to do is to incorporate more weeds, trying to have probably the most relevant weeds for the agricultural systems and this in Argentina.
And the model has a subroutine for soil temperature.
For soil temperature, for soil water potential.
And related to the quantity of straw.
Yeah. It's a question.
Just a question.
We got, what we did, we had a lot of papers, and we said, okay, we can make an equation relating the quantity of straw and which is the impact of soil temperature. And we use that equation. And we, I don't know how many papers, but we got a lot of papers, and we use the data of those papers to develop an equation. So you have different volume of straw, you have a different impact on the soil temperature and the soil water.
In the first 10
centimeters. It's better to use probably 5 centimeters, but the information we have from the weather forecast is 10 centimeters, and they cannot give us 5.
And works for a wide range of temperatures?
Yeah.
Below 0 is true?
I don't know. I suppose. No, because below 0, probably the papers we use to develop the equation don't have that information, so it's really, you know, it can be between probably 8 and 25 degrees, something like that. I don't remember exactly the range.
Thank you. Okay, we finished with the workshop. For the people that remain alive.